in Future Cannabis Project FCPO2 back in your lives. Oh, thanks guys for being here with us. We're happy to be back with you. Everybody's joining us tonight. If you're catching it live, Elka's already up this. Trevor, people love you, dude. This is gonna be fun. I already lost my spot, so thanks for joining us, guys. We're off to a romping start this evening. Yeah, as you kind of guessed it, the title gives away a little bit about what we're talking about tonight. It is the kind of the foundations of genomics, how it relates to cannabis plant breeding, and we have the perfect guest to kind of be on here and do that with us. We got Trevor, SunGrownMidz, putting in the hours today to join us today. So thank you very much, man. I appreciate that. Go ahead.
Thank you, man. Yeah, no, I appreciate the invite, Chad. Thanks for reaching out.
Yeah, my pleasure. It's a lot of people here have probably heard you. You've been all over the Future Cannabis Project, and more recently, you've even been on a lot of the clubhouse conversations about breeding here on the FCPO2 channel. And I absolutely love those sessions. And I believe it was last week. It was great. You were basically saying before all of this, you could still do this with a pencil, a paper, and a calculator. And that really, it made me think, because again, we love new fancy toys and techniques, and granted, a lot of this stuff isn't new. It may be new coming into the cannabis application, but a lot of these are established methods. And I wanted to bring you on to kind of talk a little bit more about it. A lot of us know, it's funny. I was thinking the other day, a lot of us know enough to almost kind of be dangerous. It's like, well, genomics, what is that? Well, it's the study of the genome. And that sounds pretty freaking smart for somebody who doesn't know much more than that. But it goes a lot deeper than that. And you have a great way of explaining things, in my opinion, that at least make me understand it. And I kind of almost think if I can understand it, everybody else can too. So I wanted to bring you on and really kind of focus on some of the building blocks or some of the basics. So, a person like myself can really understand how these terms are applied, how they're used, how it relates to cannabis. Take some of the the voodoo out of the breeding and whatnot. So sorry,
Yeah,
it's
no,
kind
no.
of
That honestly, I feel is something that for too long it's been kind of this mysterious black box of like, how do you do it? Like everybody gets to Mendel, and
right.
it's really simple, and it's like, okay, that makes sense. But then after that, it's like it doesn't translate, and people have a hard time bridging from the really introductory stuff that we've maybe learned in like Robert Clark's marijuana botany book to then being like, well, how does this translate to the general, like, here we are, like you mentioned genomics, and it's almost like we've been plopped down in a world of incredibly advanced technologies with a plant that hasn't been developed the same way that all of the other plants were developed, and then like genomics got applied to them, hundreds of years after there was systematic breeding, or about 100 years after really systematic breeding had already been undertaken. And so, we've got a really unique plant and a community that actually is thirsting for this information, which is really cool. Because like posting information about genetics and really getting into the nitty gritty, that's where I find I get the most feedback, the most engagement with people on different platforms. And so I think it's great to see just that the community's actively engaging in a scientific dialogue and like a process of learning and applying the science.
Absolutely. It's almost like the new toys. It's a lot of us, we want to play, we want to kind of push it further. A lot of cannabis growers were always that kid who just kind of wanted to go to the next level with everything. Um, but there are some hazards to doing this if you don't have kind of the background or the foundation, just genetically speaking, lockup, narrowing of gene pools, these types of things. It helps to have the larger picture. Again, it's just more tools to guide you. But the scientific approach, I love it. I like that level of stuff. The consummate student. Everybody is that's I was in music before this. You'll never know everything there is to know about music. Plants, no different. So I enjoy that. Um, before we get started too, another kind of cool thing you do is I follow you on Instagram. Just wanted to real quickly touch your Breeding the Culture series that you've been writing. That is very awesome. Everybody, you can definitely follow his Instagram and check that out there. But please, can you tell us a little bit about how that came about, or how you go about? It's almost like it's an awesome time capsule of historic breeders.
Yeah. It really came about from just wanting to explain a lot of these concepts and like get into the ideas of different like breeding theories, but doing it in a way that was more accessible and understandable. So looking at actual breeders in the cannabis field who have worked with the cannabis plant, the different underlying like methods or ideas that they had behind their work. And I feel like it's one of those things where learning this information gives us the tools to understand what we're already doing, because so many people are already breeding and
Right.
are already applying this stuff in a way that like sometimes our ideas have been off placed. And I mean, you guys had a great conversation on here the other night about the different taxonomy of indica and sativa and like a lot of the different issues that come up with like uh, an underground sort of counterculture interweaving with scientific concepts and trying to like use those concepts to figure things out. And like sometimes we figure it out correctly, sometimes we misapply things, but like sometimes it's not our fault. Like really, if you look at it, and like 1970s countercultures really like diving in. You got Mel Frank and Robert Clark and David Watson and these different individuals, and they go to the scientists and they look at the scientific papers and they're like, well, Robert Schultes says there's this 3 breakdown of indica, sativa, ruderalis, and like they apply that taxonomy, and then that taxonomy gets applied into the counterculture and gets like reappropriated in these different ways and like contextualized, and then 30 years later it's like, oh shit, it turns out like Robert Schultes was wrong, and it was really just an awesome human being who was trying to get his students and like people who are getting busted for cannabis off by saying cannabis indica and cannabis ruderalis aren't cannabis sativa. So legal technicality, get out of jail free card based off of this like scientist, expert witness. And I think that ended up losing eventually through the court system, but like that was the motivation, honestly. And so it's like, yeah, I feel like that's one of those things where it's really cool for us to be able to have these scientific like knowledge base and apply it. And if the science is wonky or weird, then like our applications may be wonky or weird, but that doesn't mean that the endeavor and the effort itself is like not worth it.
Yeah, it doesn't invalidate everything
Yeah,
that you're trying
yeah.
to do there. And that is it's I think that was one of the points that we all kind of came to, is just contextually, how is it being used? And yeah, sometimes there's just, what, it's the best thing we have at the moment. So until we can replace it, which is the important factor of science, is to come up with a lot of the terminology. And terminology in that context is so important, because there's so many facets to it that you have to be very specific. Um, I enjoy research papers as well. A lot of the language is dry. I make sure I have Google Dictionary next to me. But yeah, there's an absolute reason for a lot of the terminology, and that's what we're trying to get down to. And tonight too, that's what we'll be digging into a bit, is the terminology behind this stuff.
Yeah,
Um,
yeah, and it may get a little dry. I definitely, while I was writing it, I was like, wow, this is dry material, but it really does get into the historical development of essentially all of the ideas that are at the foundation of plant breeding, both in terms of like conceptual principles and then also methodological application of those principles.
yeah.
And hopefully we can weave enough cannabis in there to make it a cannabis show.
Well, I think we have, the right crowd too, because a lot of people will see how it's applied, even though it's, an agricultural study, we'll see how it's applied to cannabis, or we'll be able to relate that and to connect the dots. So, yeah, I think we have the right crowd, and if not, I'm interested. Actually, I know I've already gotten good feedback from doing this. They're like, really? Yeah, buddy. So, we're excited to have you on, but again, I keep delaying the getting into it. I just kind of wanted to ask, a little bit more about yourself, just because I always hear you spit knowledge. I never hear much about SunGrownMidz. What was kind of your background? Like, how did you get into this road? Because you've definitely dedicated, a large portion of your brain to this information. Were you growing like peas in the pea patch as a kid, or what? How did this kind of start?
I grew up, and my mom was like, she went to a hippie boarding school, and like she basically like ran away as a kid, and then like convinced her parents to send her to a hippie boarding school, which exposed her to a lot of countercultural influences. Like kids at the school, their parents rented the cabin that Hunter Thompson lived in, and so like they basically spent time up in Aspen and Woody Creek or Willow Creek or whatever it is, like in that realm. And so she like they were all deadheads and like very engaged in sort of like the post 60s. They were children of the 60s, so they were like in the 80s or whatever, 70s and 80s, and like into the hippie counterculture. And Colorado was a really like hot spot for that. And so my mom was involved in that. Always been around cannabis pretty much my whole life. My stepdad was involved in like used to import hash from Spain into Europe because he was like a British expat, and like so my whole life I've been involved in it, and it just by the time I was like 12 I was trimming, and by like 16 I was growing, and a lot of that was like I like yeah, it was a way of making ends meet and like surviving. And that was like kind of like making it through my entire childhood. Like in one way or another, cannabis basically like put food on the table or a roof over the head. And by the time I was a teenager, I like, it was just, it is what it was. Like there was, yeah. And so I was also homeschooled at that point because I got in tons of fucking trouble, like lots of trouble as a kid.
Problem with authority, or
yeah, like school. I didn't work well with the whole school system, and it wasn't like, and because I would get in trouble, then it would create like interfaces with authority for my mom, and like it was just not a good scene. I got like placed with my grandparents for a little while, and like it was one of those things where I had to be homeschooled to keep from like causing like too much shit to come down. And so I got homeschooled for all of high school. And I grew weed like as school credit, like gardening for high school as a homeschool kid.
That's pretty amazing, man. Nobody has figured out how to work that into their curriculum yet. But so it was a regular crop before you knew it as kind of how you know it today. I'd imagine then.
Yeah, like then it was just like,
chores.
Well, it wasn't really chores. It was more like, there was the it was a form of employment more, like because I was trimming and like having like seasonal work as a result of it, like
Okay.
doing labor as like a kid, like finding jobs at like places, just to be around it and to like I pretty much immersed myself because I was homeschooled and I lived on a dirt road and like there was a hippie commune down the road. Everybody on the road basically grew weed. And there was like not really much reason to go to town or to interact with
Right.
outside society. I also Calaveras County is a very conservative Republican area, and there was like these two hippie enclaves on either side of the county, and I lived right next to them for like large parts of my life, and so it was like, and we knew everybody, and like we were all friends and stuff, and so there's that aspect, and then like I kind of have an obsessive, like, if I get interested in something, I want to know everything I can about whatever subject it is. And I was always more interested in genetics and breeding and knew that that was like the thing that made the difference between like that person's herb versus the other person's herb. Like, yeah, there was some
Pretty.
technical growing skills, but like it was a
Right.
lot of times there, like the people who had the better herb were the people who were really interested in the genetic side.
That's interesting. So did you actually get to see breeding projects in practice when you were younger to kind of see the like, oh, A and B, I could get to see with a little bit of work?
Yes and no. I mean, like back in the day, early when it was like gorilla patches, there would be like a dedicated gorilla patch of just like open pollination of like a lot of
Okay.
different shit. And then those seeds would be used for the next year. But then all the other gorilla patches would be left for like your Sensi production, so to speak.
Okay.
And like that was a breeding method, and it was one that was just super haphazard and resulted in like it was just open pollination, random, and you'd see the populations move all over the place. And that's really what like that's what led me eventually to be like, there's got to be a better way. But then I also, when I read like Robert Clark, and like they straight up told me early on, like clone production wasn't really how we got down. We didn't keep clones. We were off the grid. Nobody had
Right.
power. Like the way that people reproduced their shit was through seeds and male female reproduction. And the femmes were very frowned upon. It was very like hippie organic. And so, you know, I basically like I read Robert Clark's book and thought, okay, I have to find some landrace genetics to be able to have a P1 to then be able to start a breeding program, as opposed to recognizing like that's one way of doing it. It's a very effective way when you take germplasm from very diverse places, bring them together and start making hybrids, you can get some really interesting and unique things. But that's not technically what a P1 is. Like P1 just means parental generation number one. Like that's it. It's a way of denoting a generation within a population that you're breeding. So like you take your P1 generation where you cross two plants, you create a hybrid generation, that's your F1 generation, and then you have every subsequent filial generation after that. And like the corn breeders of 100 years ago, they weren't starting with pure landrace genetics. They were starting with a huge mixed population and started inbreeding that mixed population down to pure lines and then hybridizing those and finding, oh shit, F1 hybrids, this is how we do this, and like demonstrated the science. So I feel like, I my early experience with breeding almost discouraged me from like making the dive because I was like, oh, I don't have the right germplasm to start this process. And the right germplasm is any germplasm with traits that you like, that you want to mix together, or traits that you want to improve. That's it. Like you got something and you want to improve it, then start making breeding experiments designed to try and improve it. You got something that you like and you want to keep it, breed to keep it. Like, it's one of those things where it's like, you don't have to have some magical beans that are hard to find. You can really just start with the tools you got.
And a lot of people, started with bag seed, particularly in areas where it's not okay, like it is where we are. But you kind of said just like the open pollination, and that was more of a way of just preservation. That's how I kind of got started making seeds. Again, I did not know about breeding. Breeding was not something that I set out to do because, again, it wasn't even a thought in my mind. The only thing in my mind was I'm not going to have access to more clones or seeds in the future. I was able to get some males. Why don't I start making some seeds? And that, it's again, it's a jump off point for a lot of people, but you do have to start somewhere. So I'm glad that you explained that part.
Yeah, yeah.
Yep. So yeah, let me we could get into this here. I really appreciate you kind of, sharing some of the backstory. It sounds interesting because, again, it's a plant. It's not a fast moving crop, so there's only so many chances and opportunities you get to play with it and experiment within a lifetime. So, being able to start early, I think, was probably a benefit to you. And again, that kind of helps. I'm hearing in the back of my mind, the Breeding the Culture series. You have a wealth of information. It didn't start with legalization, so that's pretty awesome, and that's a step up. So do appreciate that.
Yeah, yeah.
And so, and you've both just been kind of a self motivated, almost kind of self taught person. It sounds like when it comes to this, did you ever seek out any formal training?
Um,
Uh,
I've done I've done plant breeding seminars at UC Davis. That was like in 2015, and I went to college and just did college for a long time because I was interested in just studying stuff, and it was kind of like I can grow herb and go to college and fuck around. Like, it wasn't really like, I have no degrees. I don't have any credentials that I can point to, but I spent 8 years in college. So I have a lot of credits.
right, I got a lot of credits. And a lot of good experiences too. Again, a lot of that experience for me wasn't necessarily what I learned, but it taught me how to go about learning if I wanted to learn something in particular. The resources, where to look, the methods, structure, I
Yeah,
guess, for lack
totally.
of a better word.
Yeah, yeah. Same thing. Where research skills were learned in academic space, like being able to just spend time doing that and doing it just for personal enrichment without feeling like the grade attached to it necessarily mattered. Like, really let me just dive into subject matters that I like and to really focus on things. And ironically, I avoided math and a lot of the
Hmm.
sciences at the time and did much more history, philosophy, and the social sciences.
Hmm.
And looking back on it, I regret that choice only insofar as I've had to, like you say, kind of self-taught, go back and be like, so statistics, how do we do this? But
Yeah.
yeah, so that's definitely something that I think is a valuable tool just to pursue and do on the low key if you're not interested in getting a degree.
Right, right. Well, you don't even need a degree these days. Just kind of check out your Instagram and start clicking through some of the the links you have and folders you have up there. I know I've got one of the textbooks pulled up on my laptop right now. Actually, I am that person. I just ordered a soil science book from 1992 the other day on eBay because it was like $4 and change. I'm like, Well, it'll be funny to see what they say.
Yeah. No,
By
that's
now.
good.
Yep. Oh boy, there goes the phone. Let's see here. I'll mute myself. Nope. Okay, wife picked it up. Awesome. Good catch, wife. Sorry about that.
No.
So yeah, we let's go ahead and we can get into kind of the presentation here that you've put together. Um, and then I'll have some questions. I'll probably throw some questions at you towards the end too. But yeah, thank you for kind of sharing a little bit. I could turn this talk into a whole just 2 hours by itself because I'm
Nice.
fascinated,
Yeah.
and that's cool. That's cool. You're down in California. I thought for some reason I thought you were up in Washington, but down in sunny
Yeah.
California.
Yeah.
All right. My
Um, so should we do the share screen or how should we do it?
Yeah. Go ahead and if you hit share screen there, we're gonna figure this one out on the fly because everybody in chat knows this is Future Cannabis Project, and we do it on the fly. We do all our tech on the fly. So, and then go ahead and then you can full screen your presentation there, and then I'll be able to add it in here. So let's see here. I can go to add to stream, and we have this view, and we have that view.
Ooh, I can't see it with it up on my screen, but if we're good to
Oh,
go.
oh, yeah. Okay, you probably couldn't. So we're good to go.
Okay,
Everything looks
cool.
good there.
Sweet. So intro to plant breeding, history, yeah, blah blah blah. Uh,
Oh, please read it. It's
oh,
important. This is what we
history,
came for.
history of the scientific discoveries of the principles and methods of plant breeding and genetics. So here's like a quick intro of some of the first ideas we're going to go into. There's Mendel and his experiments that discovered the laws of segregation and independent assortment. Then we'll go into discussion on the difference between qualitative traits, which is what Mendel really researched, and quantitative traits. And this is really the bridging of the gap where people have, it's like Mendel's informative for a tiny subset of traits that aren't really applicable to what we generally do. Quantitative traits is really where the like meat and potatoes of plant breeding and genetics lies. And then we'll talk about Wilhelm Johannsen's experiments with beans that demonstrated the Caroline theory. He coined the terms genotype, phenotype, and demonstrated the gene environment interaction that produces phenotypes. Um, we'll talk about Nilsson L. and E.M. East's, Edward M. East's, and all plant breeders seem to go by like initial last name is like seems to be a convention in the plant breeding world because it seems like about 90% of people will have that naming convention. Yeah, goofy aside. Then we'll talk about heterosis, which was really like the foundation of corn breeding and the development of F1 hybrids. We'll also talk about transgressive segregation, which is a unique form of a similar type of phenomenon, but it happens in later generations, in like the F2 or later. And it's actually where heterosis is like the phenomena of hybrid vigor, where you have a essentially heterozygous genotype. It's cool, but it's not fixed. So if you make an inbred cross from that, an F2, be it full sibling or selfing, you're going to get variation in that population. Whereas with transgressive segregation, what you get is a fixed genotype associated with traits that are of a greater expression or a more extreme expression than the parent material or the F1 material. So it's an F1 hybrid with a fixed genotype is kind of how to think about it. Then Then we'll get into some of the other key ideas that were happening at the time that really kind of push the study of like experimental plant design and like plant studies, and add model organism research and the development of population genetics, which is really founded on theoretical mathematics of like essentially statistics, and also the work of some individuals named Thomas Morgan and Alfred Strudevant, who came up with chromosome theory and the linkage map. That's everything on this page right here basically shows the theoretical foundations that are necessary for doing plant breeding and were like the basis for everything that happened up to and through the modern genomic revolution. So when they developed genetic markers back in the day, they used the techniques and the data sets that these individuals used to do all this stuff.
And that's, you know, that's so huge right there. Just that slide alone is kind of the blueprint for going out and studying a lot of this. I mean, the first one that you list, Mendel's experiments, and that's usually the first one that people hear about and that they come across. So, yeah, please do tell us a little bit more about Mr. Gregor Mendel.
Yeah, Gregor Mendel was he was an Augustinian monk who, in 1865, he gave a presentation verbally over a couple of months, and that presentation also had his accompanying paper, which was then published in 1866. And in it, he laid out the results of his experiments that he had conducted evaluating the inheritance of specific traits in garden peas. He looked at 7 discontinuous traits. Discontinuous traits is another way of referring to qualitative traits or Mendelian traits. They're the traits that are controlled by a single gene. And so what Mendel was able to do through this experimental design he set up, he took pure breeding self-pollinated peas with different traits, crossed them together, and then performed a series of crosses from those F1s that he initially made to see how inheritance populations mapped over multiple generations. And that's where we get the classic laws from Mendel of the law of segregation, the law of independent assortment. It's not necessarily a law. It was more of an observation, and he didn't characterize it in such a way. But the dominant recessive relationship is also something that he identified. And he did this just by having a plan from the beginning, setting out how to test a hypothesis, and then testing that hypothesis with data and demonstrating that the plants were actually inheriting traits, even though in the F1 generation, he wasn't seeing the expression of particular traits. He was able to see them in the next generation, in the F2. And when he did the math, he was able to identify the ratios that you would get for just a very simple biallelic frequency, where you have a single dominant and a single recessive gene, so to speak. And this set of discoveries really laid the foundation for scientific plant breeding in the field of genetics. And it happened shortly after Darwin's paper on evolution and natural selection had been published. And Mendel's research was not really, it was overlooked, the significance of his experiments, because he was using simple traits in peas, and because very quickly after the discovery of Darwin's discoveries, his cousin Francis Galton developed ideas, did large regression analysis, and showed that there were continuous traits within populations. So people didn't think that these discrete traits in peas and the inheritance of those mapped onto human species. And like that wasn't even really acknowledged or addressed for about 30 years because the paper just was ignored. It went completely unrecognized, got rediscovered in 1900 by multiple different researchers. Hugo de Vries, he was a researcher who was doing research in mutation and believed that mutation was the basis for speciation in evolution, and basically ended up finding Mendel's research and seeing that it was very similar to his own. Carl Karenz was another individual who his professor had been in correspondence with Mendel during the time of his research, and he ended up finding Mendel's research. And then Erich von Teschemark was, I think his grandfather had actually taught Mendel botany when he was like at university, and he ended up rediscovering Mendel's work all at the same time. And that rediscovery laid the foundation for, you know, plant breeding and genetics, but it took about 20 years or 30 years for a really incredibly contentious debate between the Darwinian / biometricians. They were Carl Pearson and Francis Galton were kind of like the progenitors of that school of thinking. And they just did very, very detailed scientific statistical analyses of continuous variation in human populations and looked at Mendel's work and said, this doesn't work. And it wasn't until Ronald Fisher and Sewall Wright and JBS Haldane in the 1910s and 20s came around and really figured out the mathematics to put Mendelian inheritance in simple traits, how you could explain complex traits controlled by multiple genes if they were inherited through a Mendelian process, and how these continuous variations that you see in large populations are the result of the inheritance of multiple different Mendelian inheritance of multiple different genes. And so that's really like the foundation. It really feels, if you go back and read old papers from the 1910s and 1920s, it's a lot like reading old forum debates where there are people in internet forums arguing these same concepts and making the same scientific arguments that either side made. Yeah.
Wow, that's awesome. That's funny. That does make me want to go back and look at it.
Yeah, no, it's cool. Like, and you know, like academic like shade can be kind of funny, you know, like they were definitely different camps and they were pretty sectarian in their beliefs.
A lot of people, scientists and breeders, the same. We get very invested in our position, and anything that challenges it, is hearsay, or it can't be true because it flies in the face of what we think. So the Mendelian, his work was kind of met with a little skepticism because it doesn't sound like it really caught on that quick. It took 20 years.
Yeah, no, I mean, it was completely ignored, and it wasn't that it was like, it was distributed. Apparently there was some sort of arrangement between different universities and academic institutions at the time where it got distributed in a number of different copies to different places. Darwin apparently had a copy of it. It's not clear if he actually read it or not, but it's one of those things where it got distributed and nobody really paid attention because it just seemed like, well, one, he was writing, in German, so unless you spoke German, you probably weren't going to read it. And then it was also a combination of math and like he was doing statistics, and it was kind of early days for statistics still.
Okay.
And unfortunately, he was people kind of will say, oh, well, Mendel didn't, like, he just got lucky. And it's like, he didn't get lucky. He chose the 7 traits that he chose specifically to demonstrate the principle that he was trying to understand. And so he picked the right traits, because apparently in his notes he had a list of like 16 different traits. And he like got rid of all those other ones and was like, no, these 7, they seem to work in the way that I think they work, so I'm going to focus on these. And I don't understand how those other traits work, so I'm just going to focus on this to see if this demonstrates the hypothesis of inheritance. And so we're all very lucky that he did that, but it did not catch on for really until 1900, and then it became 2 very bitter camps of the Mendelians and the biometricians. And it wasn't until some people who were incredibly skilled at math were able to figure it all out and put the pieces together that it works out.
Wow. Yeah. Have a fast enough computer. Can figure it out. Cool. All right. Sorry to interrupt you there.
No, that's legit. And feel free, please interrupt me and add like, dialogue and like any input here, because this will get dry, but we are going, hopefully by the end of this, we'll have a firm grasp of the principles of plant breeding and how we can apply those in cannabis. And so like here we have qualitative versus quantitative traits. I've gone into this a bit already, but qualitative traits are classic Mendelian genetics, where the traits have a discontinuous distribution and are typically determined by a dominant recessive relationship. There can be more complex versions of that, like codominance and complete dominance and stuff like that. But typically, you'll see a very simple, straightforward distribution like this one, where there's 1 recessive, 2 heterozygous, and 1 homozygous dominant in the population distribution. And that's what Mendel identified and found in his studies for the different traits that he looked at. When you add more genes, you get more combinations. And with quantitative traits that are controlled by the interaction of multiple genes, with each one has a small effect, and the environment also influences these traits much more, there will typically be a range of overall trait expression that is continuous. And the first person to really demonstrate this was Nilsson L. And the top graph over here that shows 3 gene pairs, that maps to the actual math that he found in the gene that controlled wheat color, or the color of the kernels of the grain in wheat. And so this next slide shows the different studies that essentially establish this. First, you have Herman Nilsson L., who is the director of the experimental breeding station in Savlof, Sweden. They did a lot of very important work in the like 1800s and 1900s, establishing the pedigree method and the bulk breeding method, and doing like their pedigree records were some of the most extensive and oldest, and were able to be used to prove and demonstrate a lot of different Mendelian inheritance dynamics and populations. But he published research on the inheritance of grain color, and you can see the distribution at the bottom of the chart matches the same as the previous chart. When you've got 3 genes that control a trait, you'll see a population distribution that's like this in the F2 generation, where you've got 1, 6, 15, 20 across, same on the other side. Then after him, Edward M. East conducted similar research analyzing inheritance of corolla length, that's flower length, in Nicotina, or Nicotiana. He did that in 1916, and it really confirmed the same principles. He also talked about transgressive segregation in that paper, and it was one of the earlier papers to mention that phenomena. But both of those demonstrated that quantitative traits even though you have more gene pairs, they still conform to the same principles of segregation and independent assortment. And the dude who really like ended the debate and like finished it was Ronald Fisher. He published a paper, The Correlation Between Relatives on the Supposition of Mendelian Inheritance in 1918. And essentially he looked at human populations and said, if we take the principles of Mendelian inheritance as a given, and we look at this continuous variation in this population, here's the math that could explain how you could get to that end result. And Ronald Fisher's work in mathematics is really hard to overstate. He helped establish experimental design and like almost all of the different statistical analyses that are at the foundation of most modern sciences. He really worked out the math on a lot of those. And so he was the person who took the theory of Darwinian evolution and the theory of Mendelian inheritance and proved mathematically that they work. And ever since then, that's been known as the modern synthesis. And while it's important to evolutionary theory, it's also incredibly important to plant breeding, and his ideas on variance, his different ideas on inbreeding, and all of those different ways of doing different mathematical analyses of variation within a population are the foundation for how we're able to actually take multiple, an individual trait that's controlled by multiple genes and very uniformly accomplish significant gains in terms of the overall values for that trait in the population. So were it not for the development of the mathematics that him and some other folks that we'll talk about helped develop, this idea of developing breeding for quantitative traits would not be as effective as it is. And this was done, as we'll see, like long before the advent of, marker assisted breeding. So yeah, quick summary. Mendel analyzed inheritance of qualitative traits. He conducted his experiments in peas. He used a well-designed set of experimental crosses and math to demonstrate this. Subsequent theoretical and experimental works extended that idea to more complex inheritance of quantitative traits. That's really where the bulk of interest and the bulk of focus, if you're a plant breeder, should be. Like, it's very cool to do experiments with Mendelian traits to understand the modes of inheritance and how these like basic principles work, and for like as proof of concept. But the vast majority of traits that we like, be it biosynthesis of secondary metabolites, yield, pretty much sexual stability, any one of these traits is a multifactorial or a quantitative trait. Also, Mendel used the term factor, so early papers oftentimes use the term factor or factorial, and when they do that, they're referring to genes. That was the term that was used before this guy, Wilhelm Johannsen. Wilhelm Johannsen developed the pure line theory. He basically did a series of experiments to see how inheritance of bean size happened in a heterogeneous population of beans. And so this graph shows what his experiment was. He had a mixed seed source. He sorted them by size and then planted those seeds and tested to see how big they were and the variance within those populations. And he basically, out of that experiment, he was able to demonstrate that When a heterogeneous population is subjected to inbreeding, it segregates into separate homogeneous subpopulations of pure lines. So these individual pure lines become homozygous for the particular traits that he was studying, and that essentially he demonstrated that selection was ineffective in those pure lines for producing variants within those traits. The phenotypic variance that he was seeing when he did this experiment was the result of the different environmental effects in those seasons, and he couldn't essentially take one of these smaller beans and just create like a bigger bean, or one of these bigger beans and create a smaller bean within this population, because the traits for each one of these expressions was locked in. And there could be some variation, but that variation was just a phenomena of the environment that was happening in that particular growing season, as opposed to an inheritable trait that he could then continuously, increase the size or decrease the size based on selection. And this also reconfirmed Mendel's theory that a continuously varying trait can be inherited in a continuous fashion. So the entire population can continuously produce this range of phenotypic variation. He then coined the term gene, and later developed the distinction between genotype and phenotype, and this is where we get the genotype environment interaction. So with his experiments, he was able to demonstrate that the expression that we're seeing in observable characteristics can change while the underlying gene may not change, and we need to measure that range of variation. And that's how you're going to be able to tell the heritability of a particular trait. If a trait is only responding to an environmental variable, then it's more than likely not a heritable trait. If the trait is responding to genetic variants, then you have a range of heritability, and you can change the ratios in the overall genetic composition of the trait.
So real quick, because genotype is one that sometimes garners a lot of confusion for people, because genotype, sometimes people think that it is locked in. The gene is the gene, it's going to express itself the same every time. It's the phenotype where you get the variants or the expressions. But with this variable of the environment, it sounds like there is actually a little plasticity to genotype, whereas my environment, your environment, the same genes will still be a little bit different. Is that, am I kind of framing that correctly?
So he's basically saying genotype is constant and not changing when you're dealing with a pure line. So when you're dealing with a truly homozygous individual for a particular trait, and that trait is, there isn't any variance in the population for that trait. Every individual has the same genotypic variance. They're all homozygous dominant, let's say.
Yeah, and we can just say it's purple.
Okay, yeah, they're all purple. So that purple as a genotypic expression, let's say it's homozygous, it's a fixed trait, that is something that you're not going to get any additional purple because of the genetics. You can cross those plants in that population or self that individual, and you're not going to get any increased purple. But you may one year get a more extreme expression of purple because your diurnal temperatures are much colder, and purpling is a phenotypic expression that is at least in part related to an environmental characteristic of cold temperatures. So in an environment where you've got colder temperatures, one season, you may actually have a much more dark and extreme purple expression. But then in the next season, when it's much warmer, that environment is less conducive to the expression of purple traits. And so as a result, even though you've got the exact same underlying genotype, the next season's purple expression is nowhere near as extreme as the other years. And when you like measure those and evaluate them from year to year, like if you were to do a breeding program for a purple specific like purple end goal, you would want to score your trait on a continuum of like not so purple, kind of purple, yeah, okay, mid purple, ooh, that's purple, fuck, that's purple. Like, and so like +5 is like fuck,
Right.
and that's what you want. But if you're selecting just on that, you're not accounting for the environmental variables there, and you see the 5 and you're like, oh yeah, no, it's purple, it produces just like the deepest, darkest purples every time. But that was just a phenomena of 1 year's environment or maybe multiple years environment, and not necessarily the result of a fixed genetic variable, or a not necessarily a result of a genetic variable as much as a environmental variable, which produces variability within your phenotype. And so, and like each one of these creates different levels of variance. So you can have genetic variance, you know, homozygous, heterozygous, or additive, non-additive. When you're dealing with more quantitative traits, you're not really dealing with dominant, recessive as much. You're dealing with a more complicated math scheme of variance, which is something that Fisher developed. And so in that situation, your genes create some degree of variance. The environment creates some degree of variance. The interaction between those two then creates another level of variance that can be in addition to just the genetic variance. So you can have genetic variance, and when they do the equations, they have ways of quantifying each of these different levels of variance and interactions between different types of variance. And that's what produces your phenotypic variance. It's the complex interaction between not just gene and environment, but gene, environment, gene X environment. It's like the equations, when they write them out, have an extra bracketed level of complexity based on the interaction. And that's really what, had Wilhelm Johannsen not developed these theory, or like done this experiment and presented all of this, we wouldn't have that conceptual apparatus. And like every study subsequently that has really looked into and refined these ideas has kind of stemmed from this initial conception of the distinction between the expressed phenotype and the genotype, and then trying to figure out what's the variance at play there.
Awesome. Thank you. Yeah. So it does sound like just kind of, you know, again, please correct me if I'm wrong again. But the genotype is kind of fixed then. It is what it is with some variance to it, as you're saying. But that environmental factor is more of, it's either going to amplify the effect of the said genes or turn it down.
Yes. Yes.
Okay. Okay.
Yeah.
It's not going to.
That's really a great way of framing it, because that's what happens if you have upregulation and downregulation for particular things. And so, you know, like if you're growing herb in suboptimal conditions, you're not going to have upregulation for all of the biosynthetic pathways that you're trying to upregulate for to produce the terpenes, to produce the cannabinoids, because you aren't providing the environmental inputs that the plant needs to express those things. And so a lot of times, all like the genotype they set parameters, whereas the environment then determines the range of expression within the parameters created by the genetic material, essentially. So like if you don't have the genes for, you know, some minor cannabinoid or for high ratios of a particular cannabinoid, you're not going to get it by just crossing that population together. You got to add variants into the population through genetic variants. And, you know, there used to be ideas that you could just take a variety from X location and then move it to another location, and that change in environment would then result in higher cannabinoid production. And or if you take it up into a higher elevation, you're going to get higher cannabinoid expression, and that's going to be an inheritable trait. And that doesn't appear to be the case. Like, you may get a higher expression within the relative parameters that are set by the genotype. You're not just going to be like, hey, I got high THC fucking cannabis out of nowhere, when you're rocking like true industrial hemp genes. You know, like that kind of mutation is rare and is not like, and that's what that would be. That would be a mutation. And mutated genetic variants through mutation is a very rare thing. It does happen, but it's not like, it's not very common.
Don't bet the farm on it.
Exactly. Yeah.
Okay.
And so yeah, that was Wilhelm Johannsen's pure line theory. And like, really, this kind of showed that you go down a particular avenue, you can fix a trait, but you may not be able to get increased expression within the trait if you just go down that single avenue. You know, if you fix the weights, the size of the bean, you're not going to magically start producing bigger beans by starting with material that has a fixed genotype that that doesn't have that within its genetic range of expression. So then these cats, George H. Schull and E.M. East, independently discovered the phenomena of heterosis, which is hybrid vigor. And essentially they took inbred lines. They kind of built on this idea from Wilhelm Johannsen of taking an inbred line and making just separate pure lines out of it. And by intentionally selfing over multiple generations, because corn's a outcrossing species, but it also it has like a weird ratio of selfing and outcrossing because it's a windblown species that has it's dioecious, so it pollinates itself, but it also is receptive of other pollen sources. And so they intentionally, through mechanical interventions, created inbred lines. As you can see, these inbred lines are very small relative to their hybrid offspring, and that was largely a result of the inbreeding depression that gets experienced when you start fixing deleterious recessive. It's usually attributed to recessive traits, but when you inbreed long enough, you start seeing a depression in the overall yield and expression of the traits. But what they demonstrated was that if you then cross these together, you produce hybrid offspring that exceeds the trait expression or the yield for the parental material. And this was really revolutionary because breeders had been using a open pollination mass selection process for selecting corn for years and years and years. And this graph right here shows, right in this area, there was essentially zero gain in yields from 1860 to 1920. And that was because using mass selection in an open pollinated crop wasn't actually, they weren't able to get the increases in the quantitative trait that is yield just by this open pollination process. Instead, they had to work these inbred lines and then develop the hybrids, and it took a little bit of time because at first they weren't economical, and they did double cross as like a intermediary stage, because literally the inbred lines didn't produce enough seeds to like plant all the corn across the the country and or world. So like they just didn't have enough actual seeds produced by the inbreds and the hybrids to, like, when you take two inbred lines and then make the cross, they weren't able to get enough seed to meet the demand, and it took them a number of years. And a lot of it had to do with the labor costs of detasseling. And so once they got cytoplasmic sterile lines of corn, that's really here on this line. And that's when F1 hybrids from single crosses of inbred lines became the norm in plant or maize breeding. And like these yield gains are fucking insane. Like it's pretty incredible what plant breeders were able to do, and there were all sorts of drawbacks with this. This is definitely a resource-intensive method of crop agriculture, and also, the Chimera talked about it a couple weeks ago, but they basically, by taking this approach, ended up breeding in a susceptibility to a blight that ended up wiping out corn production in huge areas.
Yikes.
Yeah.
Yeah, monocropping. That's a different subject.
Exactly, yeah. But like this is like where we're here. Yeah. Like
with cannabis or just in general? Okay.
Yeah, the cannabis like population, and like I mean we've gotten some gains like this in terms of particular traits. Like I'm sure if you were to map THC in particular, you would see THC concentrations that would map on a graph somewhat similarly to this. We've had an enormous increase in cannabinoid concentrations. That said, we're still dealing with a largely open pollinated population structure that there's been selection for particular traits, and that trait architecture is definitely like, we see it, it's pretty ubiquitous. But because it's been this process of introgressing whatever the new is, and like constantly being like, okay, we've selected this thing, and now everybody's going to use this thing.
Right.
And doing that for 30 years makes it so that, like, we've got a population structure here, and if we start doing the inbreeding and then doing the hybridization, particularly with cannabis being an obligate outcrossing species, which we'll get to in a little bit here, but like that reproductive system makes it so that hybrid breeding is really kind of the end goal, and not the end goal. It is the commercially most, like, it will put you on this kind of trajectory and make viable commercial products that can be grown in large field agriculture and can be monocropped essentially. And that will be economically boon to certain individuals and corporations that do it.
Yes.
That doesn't mean that that's the only way. Like, that is one way, and it will produce incredible gains, but it has incredible drawbacks. And that's what I kind of was getting at with the Chimera reference. Like, this is one model. It's not the only one.
Right. Do you think that inbreeding depression has anything to do with why we may be at that low baseline right now? Or does that kind of play into it later? Because
no, that definitely plays into it. And that's like, we have a weird, It's like we have inbreeding depression, but we've ameliorated that by constantly introgressing new things and like outcrossing it so much so that we have a highly heterogeneous population that's just been skewed for particular traits. So what we have is mass recurrent selection for cannabinoid profiles, some solid selection on terpenes, but largely that's a crapshoot. And then like yield and like flower time, like those are the primary traits that people have really skewed the genome of the plant and kind of developed some bottlenecks in terms of trait expression. And on some levels, it's good because we're seeing some skewed higher expressions.
Mm-hmm.
But what you're going to see is that you're not going to have as good gains in those traits as you will with other things. And where we haven't really like fucked around is yield. And so like we're down here in yield because we've got all these different traits, but we haven't put them onto a thing and uniformly established like, okay, this is the trait architecture and the plant structure that produces the best yields and the best in this like regional agronomically important area, like the Central Valley of California or Mendocino or
high winds,
you know,
dry,
like,
yeah.
And so that's something where like there's for most other crops, like there's a corn belt that like stretches the world. There's a cotton belt. There are like particular areas that these crops are grown in based on their environmental characteristics. And the crops are bred to do best in those environments. And they've been specifically like the characteristics of the plants as they're being like developed and like brought to market are for the environments that they're being produced in. And like with us, we haven't even begun to scratch the surface there.
Yeah, we're just starting that conversation of you know, again, I my goal is to get things for people in tents. Low ceilings, indoors, limited space, something that's going to put out there. But to kind of play off what we've already been learning earlier, you know, the yield and stuff, the yield that you had mentioned, the kind of the structure you mentioned, those are all qualitative traits that would kind of fall under Mendel's dominant recessive because we're able to physically see this one yielded more than that one.
Well, no,
trait?
They're quantitative because we can look at all the yields on every single plant and be like, okay, this one was like way down here on the low end. This one's way up here. And when you map all of them out, you're probably going to see a distribution that's closer to a bell curve. And it's a much bigger range of variability, typically with yield, than there is with like, typically a qualitative trait is a yes-no thing. So is it purple? Is it not purple? Is it autoflower or is it photoperiod? Is it the mutants are great examples as well.
So you don't need data really, like you said. It is the yes no. Whereas yield, yeah, I guess that would be a better way of tracking it. You know, I'd rather have data from 1,000 plants versus the 20 I could see physically and be like, yeah, well, these are the yielders over here.
Yeah, and you'll usually see that like, or like with that type of breeding, essentially what happens is every generation you doing this phenotype analysis and the tracking the data on yields. And so you'll be like, okay, the parent population has this continuum for the yield and the mid-parent like yield expression is X. And then the next generation, you'll grow out the population and you'll look at the mid offspring. So like, what is the mean for the parents? What is the mean for the offspring in terms of yield? You compare those And that right there is the response to selection. And that will tell you the heritability or the response to your effort to improve that trait. So if you get, the mean for the parent on yield is like X, and it then increases to whatever in the next generation to Y, well, then you've demonstrated that your selection in that generation was a good positive selection for that particular trait. You've improved yield, let's say. And that's every generation you're doing that. And like that's how traits are improved on the quantitative level. And a lot of that is establishing like a threshold where you're like, okay, we're going to take the top 5 to 10% of yielders in this generation. We're going to keep the seeds from those, and we're going to propagate them in the next generation and keep doing that. And that's typically like the intensity and the accuracy of your selection is oftentimes a function of the threshold that you're creating for like how many of the population are you going to use to create the next population.
Thank you.
And like this graph kind of shows a crazy example. So this is transgressive segregation, and what you can see in this picture is this right here is one of the parent plants. This right here is one of the other parent plants, and this is tobacco. They hybridized those two plants and produced this plant. And you can see that there's clearly a gain from here to here. Like this one's relatively equal, but like they get a good gain on relative to one of the parents. And this is an F1 hybrid where heterosis is being exhibited relative at least to this parent. In the F6 generation, so this is after 6 generations of inbreeding, this is the transgressive segregant that emerged in the F2 population that they just continued to inbreed that far exceeds all of the other, its parent and grandparent populations. This is how you can significantly increase and fix particular traits. In this instance, it's vigor and yield. So transgressive segregation, like I said, is a similar phenomenon to heterosis, but it happens at later stages. So this quote from Goulet in 2017, quote, transgressive segregation is distinct from heterosis because it manifests predominantly in the F2 generation and later and may persist indefinitely once established. This shows that, like, this plant was a transgressive segregant in the F6 generation, but it had already shown itself by the F2. And it was by selecting the transgressive, the one, or not one, but like by selecting the individuals, there may be more than one, that are the most extreme in phenotypic expression that exceeds the parents, is essentially within the like F2 population, is a transgressive segregant and is something that is worth, one, identifying, and two, breeding with, because you've essentially fixed the genotypic value for that trait and it's inheritable from that line. You can, you keep selfing it, it's going to continue to produce that trait, and you're not going to get variance within that trait. And like this should be exhibiting significant inbreeding depression, but it's not because of the transgressive segregation dynamic. And it's a very complex, like essentially they think it's a complementary gene action combined with epistasis that makes it so that even though you have a homozygous genotype, the overall gene interactions produce this extreme expression. And these are the different papers that look into it. It was predicted by E.M. East and described by him. J.P. Lotze wrote a book on evolution, and it was then more like researched in the 1950s and really kind of like empirically demonstrated by H.H. Smith, who was the person who did the research in 1950 on tobacco. There's research into tomatoes on the transgressive segregation. And if you go into the forums or you read anything by DJ Short, if you read anything by Tom Hill, they're oftentimes talking about the phenomena of transgressive segregation and how you want to have your selection frequencies at 5 to 10% of a very large population. You want to track that population for the traits that you're most interested in, particularly for these quantitative traits, and a great deal of like detail, so that you can be like, okay, here's the threshold. Like, these plants are the top 10% yielders, or the top 5, or if you want to be extremely selective, the top 1% of yielders. And that's how you're going to be able to track transgressive segregants and also track the response of the traits that you're selecting for from one generation to the next. Chromosome theory, we talked a little bit about this with Chimera on the breeding roundtable, and it was really interesting to hear him talk about just how chromosome theory emerged, and also how Alfred Strudevant was a 19-year-old student who literally just took home a huge stack of data from fruit fly breeding experiments, and while looking over the data, identified some patterns. And essentially what he did, he was able to calculate the crossover rate for traits within a population, and they had mutants. So they had a white-eyed mutant that seemed to be a male sex-linked trait. Because they had that one individual that showed up with a mutation that was a recessive sex-linked trait that made it so the offspring, I believe, females, had white eyes as well, he was able to track that trait throughout the population. He was then able to track the other traits that the population was also had. And through that, he essentially did the math and found that it happened in a very unique, or a very predictable and consistent pattern. And the closer a trait was to another trait on the chromosome determined the likelihood that a crossover event between the two traits happened. And because it was so consistent, you could actually map it. And essentially in 1913, he created the first genetic map. So when people talk about genomics or, marker-assisted breeding, or, understanding the actual, like, where the placement of a trait is in relation to another trait on the actual chromosome, the mathematics for figuring that out and developing those ideas were first developed by a 19-year-old in 1913. So when we talk about like having a pencil and paper and a calculator, like that literally is how this stuff was figured out. And, like, granted, I personally lack the statistical background to like do the math, but I know a guy,
Right.
And the internet, like the cool thing about all these things is now that they exist, we're able to a lot of times input these formulas into, like, we can input data into a formula that already exists in an internet like website that you can find, and you can figure out these types of data, or these types of like This one's a little unique. I mean, genetic mapping is very like involved, and they literally spent like the better part of the 1900s like mapping all sorts of like mice, maize, barley, and all these things in really extensive detail. And then like in the 1990s, genomics came around, and they're like, "Fuck, now we can do this in like 10
Oh
minutes."
no.
Yeah,
Yeah. Well, you got to start somewhere, but that actually really does blow my mind that it goes back that far. Um,
yeah, me too. I was the same thing. Like we got dropped into the genomic, like, you know, I grew up in a post DNA world where, like, you know, CSI, blah, 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 like,
yeah.
a
Right.
DNA, he's guilty or some shit. And,
Yep.
like,
Yep.
it's crazy to think that it goes back to the early 1900s and that it was really just people taking huge amounts of data and analyzing it for patterns and then being able to translate that into actual material reality. Like, here's how this math shows how chromosomes are the mechanism on which DNA, and they didn't have DNA at that point, but like they knew about genes and they knew that there were these chromosomes, and they're like, okay, these chromosomes are doing some shit. They're carrying these things, and these things, genes or alleles, seem to be ordered in a very like systematic way. And that's something that we can, you know, all we have to do is recognize that if we take data and apply the techniques that have been developed by these individuals before us and use the equations that are here on the internet, we can actually do all this shit ourselves and can really, like, as a community, apply these tools in a way that we maybe haven't, you know, individually. It's very interesting the way like these things work, because there are certain individuals who are like way ahead of the curve and have this shit figured out and have been doing it for years, and then some of us are like, wait, what? Like, I want to fuck around and find out too.
Well, it sounds extremely complicated, but I guess once you get the formula, you kind of you see the big picture. But that is kind of mind blowing that something that big can be used on an individual level, and that's actually practical to be able to do that, to steer something that a lot of us are trying to do. So,
Yeah, yeah, and that's like that's something where I feel like when we stop at Mendel, or when the discussion stops at like it's very simple inheritance of discrete traits, it ends up missing the, well, yeah, it's a more complicated discussion, and yeah, there's a lot more math, and yeah, that's kind of why I stopped going to college. But like, now that I love cannabis and I am actually interested in variation within a population, and okay, fuck it, I'm gonna like dive in and try and learn statistics, and not just statistics, but like the history of the development of these ideas and how I can practically utilize them in my own life. And so like now I'm all like, it's my jam. Like, I
yeah.
fucking love statistics and math and just looking at variance and analyzing data. Like, data is something that I now find very interesting, and a large part of that has to do with just learning about breeding as like an entry point into mathematics.
And the practical applications. Yeah, definitely. It is funny what lengths you go to learn or further your knowledge when you're interested in a subject. I'm
Yeah.
not gonna claim to be like the great, youth student or high school student or anything like that, but when it came to, not too long ago going back to school for the sustainable ag stuff, motherfuckers on the honor roll, like straight A's. Like, I'm like, where did this happen? It's like I called my mom right away. I'm like, look at my report card. So, yeah, I hear you there.
Totally. Yeah. Yeah. And that's, I mean, you know, it's one of those things where a lot of us, like people who got into cannabis in our, it's you know, prohibition made it so that like we all learned about cannabis either from our friends or at a very early age. I happened to be like in it, and like I honestly, it was when I met as a like 12-year-old some other kid who like was a suburban sort of like normal person. And he was like, "Oh yeah, steal that shit. We can get high." And I was like, "Wait, what? Like,
What?
I'm into forts and shit. Like, oh, fuck you. Oh, well, okay, I'll be down." And that's when I realized that you can get high. And then once it was found out that I was getting high and shit, it was like, well, fuck it. Like, okay, like you better make some fucking trimming money sort of thing out of it, and like stop stealing fucking people's weed, dumbass. Was kind of like the vibe on it. And so like for me, I feel like so much of the counterculture was kind of like anti-authoritarian, anti-system, and so like a lot of that manifested in my early years of being anti-school. But now, like, I fucking loves that shit, and like, you know, it's why I did 8 years of college. Like, this type of information is something that makes it so that if we have this knowledge, we can apply these concepts and we can understand like where they come from without it just being like, you know, something that feels like, oh, you have to have millions of dollars or some academic training. Like you really don't. And like you talk to a lot of academics, and they're very surprised at how conversant and engaged and knowledgeable cannabis breeders are, even if cannabis breeders don't know the the exact terminology. The fact that they're engaging in plant breeding and have experience with making hybrids with populations, they're oftentimes they're more like, wow, the stereotypical like dumb stoner is like disabused immediately, and you get into some really great conversations with academics in the plant breeding space in particular. But yeah, so that's kind of all of the stuff we just went over. Last theoretical idea, population genetics. This was really the foundations of, this is like the next level shit, where they really started to understand all of the different mathematics for understanding like population dynamics. And it first started with a very simple equation. It was found by two different people. It's the Hardy-Weinberg equilibrium. And this idea is that when you have a population that's a very large, completely random mating population, you're going to maintain a constantly allelic frequency. So if you just use the traditional Mendelian sort of inheritance and trait expression, you're going to have the homozygous recessive, the heterozygous, the two heterozygous, and then the one homozygous dominant. And that type of allelic frequency will continue throughout the entire population over generations. So if you don't have any environmental natural selection, if you have zero artificial human selection, if every individual in the population theoretically can mate together, and there aren't any barriers to reproduction, then, and I think there's one other, there's like there are 4 requirements that are theoretical, but that will maintain a population in equilibrium. And so this was important because it was kind of demonstrating that, like, yes, you could have a distribution of variance in a population that maintains, like, stays constant, even though you have different types within the population. So you can have the homozygous for dominant and recessive and the heterozygous, and they can stay in a constant frequency in a population, assuming all things are equal and you have random mating. That's not what happens in any population, and they fully understood that, but it was essentially a way of demonstrating mathematically that Mendelian principles were at least theoretically possible in a population that maintains some consistency, because that was kind of a question with the synthesis between Darwinian theory and Mendelian inheritance. It was kind of like, well, if that's the case, then you're going to have speciation and variation that leads in divergent tracks that happens way too much, and we just don't see that. And so they demonstrated that the consistency that we do see in terms of this distribution of frequency is a product of math. And then Ronald Fisher, I've talked a lot about him. He really is one of the people who puts statistics on firm theoretical grounding and applied it to tons of scientific applications. We use analysis of variance for quantitative trait inheritance analysis. Experimental design is a way of checking for environmental variables or different variables that might bias your research results. So if you actually look at breeding programs, they're typically done on a plot design where there will be however many different plots with X number of plants in each plot, and there will be a check at certain intervals, and essentially they'll be able to, through that map, and determine whether or not variance that you're seeing within the trait expression is the result of placement within the experimental design or within the actual environment, or if it's a product of some genetic variance. And so he was really important. He also is like, he was a bit of an asshole, and definitely like had some controversial views, and was like, him and Sewall Wright, another population geneticist, like they went hard at each other. And
oh no.
Yeah.
They weren't invited to the same fundraisers.
Yeah, no. And then JBS Haldane was an evolutionary biologist. He published 10 papers that really put Mendelian inheritance and Darwin's theory of evolution into the same thing as what Fisher had done before him, but just looked at other traits and demonstrated mathematically how Mendel's theory of inheritance applied to all these other traits. He also did the first genetic linkage maps in mammals and in humans, and he's the guy who developed the rate of mutation. He was the first person to calculate that and recognize that there's kind of a uniform rate of mutation that happens within the genome, and that you can kind of like, that's now used for regression analysis in genomic dating. So like when they say, oh, this comes from X time period, usually what they're doing is they're analyzing the rate of mutation of the genome over time and using that as based off of Haldane's work to say, okay, this is how much time period has elapsed from, when this population either existed or like now, sort of thing. This was brought up, I know, for that the recent study out of China talking about the early days of domestication. That was a molecular clock analysis that was based off of the rate of mutation.
Okay. Yeah, I did read that one. That was interesting.
Yeah.
Trying to, again, answer the age-old question, where did it all come from?
Exactly. Yeah. And that's like, and there are different ways of doing that calculation. And so that's something that can, like, it creates variability in those dating, and also becomes like, the experts will debate over those sorts of things, but really the person who that tracks back to is Haldane. And that's also how, like, they developed these ideas of evolution, and like for a long time it was like, is mutation the result of speciation, or is it some other variable? And they found that like it's some other variable, but it was largely through his work. And the last population geneticist is Sewall Wright, and this dude is very, very important to the whole breeding world. He developed the concept of the inbreeding coefficient, and so like when we say like selfing increases homozygosity by X, we figure that out by using the mathematics that he developed for checking what using a pedigree chart and the lines of descent within a pedigree chart, you can essentially trace back and identify what the probability of homozygosity for an individual in a population is based off of their pedigree chart and their relations. And he demonstrated it for selfing populations, for full SIB populations. So that means like the quote unquote traditional F2, where you have a male and a female sibling and you cross those two. That would then produce a inbreeding coefficient of 0.25. So essentially, if you have a population where you have a homozygous and a homozygous parent, so two homozygous parents, you cross them, you create a heterozygous offspring. You then take the heterozygous offspring and self it. You're now going to have an inbreeding coefficient of 0.5, where 75% of the ensuing offspring should be homozygous. 25% should still be heterozygous. If you do it with the siblings, you're only going to get it has an inbreeding coefficient of 0.25, so you're really only going to get increase of like 60%, as opposed to the full 75% that you'll get for the selfing and back crossing as well.
So, well, no, actually, okay, if we're talking selfing, I'm just trying to think. I don't know if you know the story of Cindy 99 and Mr. Soul and how we got to
yes. So this is a great example of this discussion.
Okay.
But yeah, go on, like lay that out.
Oh, well, yeah, because as you were talking, that was kind of what was coming to mind, because I've liked that idea of being able to have something that's super elite that you like, but being able to get it more like that down the road by adding in something that is easily identifiable and not elite. And then you keep going down, and you were hitting the numbers, but then you also said selfing, and I'm like, well, no, I don't remember selfing being part of his process.
Yeah. No, that was like, there was beef back in the day on the internet between the selfers and the back crossers. But it was definitely Mr. Soul, Vic High, and a number of other people were very, very big proponents of the idea of doing back cross breeding, and it was presented as a very simple, straightforward process of after 4 generations, or quote unquote cubing, that you would essentially take a population where you went from a initial generation of the hybrid of a 50-50 split, then you would increase that by 25% of your recurrent parent. So you go back to the Cindy 99, let's say, or it was Princess, I think. So you take your F1 offspring and you breed that back up to this recurrent parent. So now that offspring is supposedly 75% of the parent's genome. Then you take that and you take the offspring from there and breed them back to the parent. You do this for each generation, and every time you get like 25%, 12%, 6%, 4%, etc. gain, or 3%, excuse me. All the way to 99%. And then in theory, by having made these back crosses to the recurrent parent, you'll have 99% of the parent's genome after about 4 or 5 generations. Theoretically, it's true. And Seawall writes inbreeding coefficient is the basis for that idea. But there are complications, and that has to do with linkage. So when we talked about Strudevant, and he was able to calculate the distance between different traits on the actual chromosome, when the chromosomes are inherited during meiosis, the crossovers that happen, if the traits are really close to each other, there's typically not a separation between the two traits. And so they're inherited together a lot of times. And so while theoretically the inbreeding coefficient would result in, here's the numbers. If you have a highly, highly inbred recurrent parent, so not princess, but like a super inbred recurrent parent, and you inbreed it for multiple generations, this graph shows right here what that would be. So you start out here, you make your F1 hybrid, you have a heterozygous genotype. You then take this and you backcross it to the parent. About 75% of the population is going to be demonstrating the parent, like the homozygous parent traits. Again, you get to 87, 93, 96, 98 by like generation 6. But if you're dealing with highly like repeated backcross to a random bred individual over here, so you have a not a homozygous, not an inbred line, but just a random line, these are the kinds of inbreeding coefficients you're working with. And as you can see, by generation 10, you're basically at 50% recapturing the genome.
Oh, wow. Because that's usually, if you're starting today, as far as cannabis, that's what you're starting with.
We're
is
starting
column D. Okay. Yeah. Interesting. That's awesome math right there.
Yeah, this chart's really, really helpful. And it shows different breeding systems. So here you've got the full brother, sister, or the offspring younger parent. And apparently in like a lot of crops, the younger parent creates a different variable than the older parent when you make inbreed, like do this inbreeding method. I don't exactly understand the principle there. I haven't researched that one, but selfing, this chart also shows the selfing inbreeding relationship, and this is another way of expressing it. So when you use selfing for inbreeding, you're going to increase the amount of homozygosity to 50% in the first generation. Then by the second generation, you're getting to like 75, up around 80, still in the 80s, and then into the 90s by generation 5 to 6 and 5, 6, 7, 8. And this shows the actual breakdown of the genotype. So here, generation 0, you have a heterozygous population, heterozygous for this genotype. Then you inbreed it, you self it once. Now you have a 50% homozygosity to heterozygosity. Then here, and you can see even the numbers that you would have breaking down on the fractions in the actual genotypes in this graph. And so here you've got 28 homozygous to 8 heterozygous to 28 homozygous. So what very quickly starts to happen is your population starts to differentiate into this extreme over here and this extreme over here, while this starts to reduce in number. And so like by generation 7, you're getting 8,128 homozygous for both dominant and recessive, and only 128 heterozygous. So there is that variability will still continue to persist, and you'll get to 99%. But this is after, 7 generations of selfing, which is the most intense form of inbreeding. And what this rate of heterozygosity is, this little fraction over here, is referred to as residual heterozygosity. And even going 20, 30, 50 generations down the line, they've found that there will still be some tiny residual amounts of heterozygosity even within extremely self lines.
That's awesome. And we got Peter Peters joining us in the chat. He's actually saying the little present button in the upper right hand corner next to the yellow share. It's like if you press that, then it might actually, or the present button.
Oops, my bad.
Yeah. No, that's okay. Yeah, I'll take it off full screen here. Let's see. Oh, bump. Okay, that happens. Sorry to throw you off there.
Oh no, it's all right. I'll get it.
It's all good.
Present.
And I don't know if this makes a different view for you. It's kind of the same. Let's see here. If I go to shoot full screen, yeah, it's a little bit bigger over on this end. I don't know how easier it is for you. You could go back to the old way if it's easier too.
Oh no, this is fine. Sorry to the. Okay. So yeah, we just did there. Yeah, we're good. So, yeah, and this is where like we've already kind of jumped ahead, but these are the ideas here. And like when Chimera was on the breeding panel, he kept stressing the importance of understanding the reproductive system of cannabis, and that's something that I definitely think is important. And there's a reason why, even though it seems kind of rudimentary, like it's fundamental to the process, and understanding that is really key. And so like these are the basic reproductive systems that you have for the species. You've got outcrossing, which is cannabis. Cannabis is a naturally outcrossing species. You've got selfing species, so like barley or a lot of the cereal grain crops are selfing crops. Peas are a selfing crop. And these are plants, they're autogamous. They pollinate themselves. 90% or more of the plant kingdom is monoecious, meaning that they produce both sex parts and are able to pollinate themselves, typically speaking. Cannabis, where you have a dioecious species that's wind pollinated, it's able to do lots of intercrossing within the population. Asexual reproduction is another form of reproduction. Certain plant species also have this, like potatoes, tubers, different things like this have ways of reproducing themselves. This is a unique form in like the actual, like in nature. Cannabis isn't an asexual reproducer, but we are able to do asexual reproduction through cloning and micropropagation, and also selfing through chemical manipulation. So each one of these is applicable to cannabis, even though it's an obligate outcrossing species. And each one of these types of reproduction then has implications for the species. If you have simply a system of asexual reproduction and we're completely dependent on clones, and then, I don't know, a fucked up virus shows up and starts wiping out crop production like hop latent, that creates a crop species that is very much at risk of pest and pathogen pressure, and a industry and production that is put at risk because of these types of pressures. Selfing with an outcrossing species results in inbreeding depression, and so you'll get more deleterious outcomes in terms of the population, but that can be offset by things like heterosis and the fact that you can use those inbred lines as breeding material. But it's like, there are without a doubt some genomic consequences of choosing a path of extreme inbreeding. And it can result in just individual plants that may not be as productive, plants or families that may not actually continue to be viable because they're simply not able to survive. They'll either be infertile or they won't, yeah, they'll literally die off. And those are forms of, there's a, I always forget the term for this one, but it's, there are barriers to inbreeding, essentially, and in outcrossing species, and they can be greater or lesser. And in cannabis, it's an intermediary. So cannabis is a dioecious male-female species. It has male and female sexual organs on separate plants. It's anemophilous, so it's a wind-blown pollen. And this came up on the breeding roundtable. Apparently, I was looking at some studies, and Camara had mentioned that in Spain and France, they're able to estimate the size of the Moroccan hash crop based off of pollen that flies and like blows into Spain and France from Morocco. I believe those are the studies that have stated that wind-blown pollen can move up to 300 kilometers or kilometers. So that's pretty enormous area, and as a result, cannabis is in its natural state is just incredibly diverse and heterogeneous. You have the possibility of different populations that are separated by hundreds of kilometers interbreeding. And so that's something that our cannabis is typically a very heterogeneous, very heterozygous type population, and the overall population health is typically in that genetic state. So when we put it into an intensive inbreeding program, you're going to see a lot more recessive, deleterious outcomes, because those recessive alleles, while they may be deleterious in a crop, like in an intensive agricultural setting, they create plasticity within a heterogeneous population. So they may produce suboptimal outcomes on lots of years, but when the environment shifts or when different variables shift in the environment, it makes it so that the population as a whole now has more diversity to be able to adapt to new environmental conditions where it may have greater fitness. So recessive, like deleterious alleles can exist within these type of populations, whereas in selfing populations, they've already done a lot of the genome cleaning, so to speak, of getting rid of these deleterious recessives, because, and there are also versions of deleterious dominant, but like most of these are recessive traits, and those get weeded out in a lot of selfing populations where they don't necessarily get weeded out in outcrossing populations.
So I'm not necessarily say that you're picking a side on this argument, but if somebody was making the argument that selfing has its advantages, that sounds like one of the advantages it could add to its list.
Yeah,
Versus an outcross.
Yeah, and I mean, you're gonna in selfing, and one of the reasons why it can get a bad rap is that you are going to see deleterious outcomes, and you are going to see plants that aren't very viable and don't produce necessarily what a commercial producer is looking for. But that process of getting rid of, like, on one level, you can do selection against those deleterious outcomes, and on another level, those deleterious outcomes will start to cull themselves as well, which is something that you want to do. Like in your breeding programs, having undesirable recessive traits is not something that's necessarily good. And if it doesn't affect fitness, like the mutation breeding may improve fitness in certain environments. It appears, I know High Grade Mutants has posted a lot of stuff about leaf morphology and how Cannabis Research also is another one of the IG handles. but he's talked about how he's got the Freak Show line that he's been working with for years, and it has a really unique fern-like leaf morphology, and how that leaf morphology, while it may not be the best leaf morphology for a plant that's out in full sun where it's able to get the full advantage of direct sunlight, it may actually help to have that more very like intricate leaf pattern that is able to then catch the patchy sunlight that's coming through a canopy, because it's not wasting a bunch of energy on this massive leaf that isn't actually ever going to get direct sunlight and acting like a big massive solar panel sort of pattern, but is instead just going to catch these little spots here and there. And so it saves on overall energy by doing this leaf system. Now, that could be a totally beneficial trait in certain systems, and then not so much of a beneficial trait in other systems. And the fact that we have that is a good thing. Like, that adaptability for the species, like, who knows where we're going to be climactically 30 years from now, 100 years from now. And so having that built-in mechanism is a good thing. So where selfing can, on one level, get rid of recessives or eliminate deleterious traits, it can also fix and stabilize and maintain those traits as well, for that may be adaptive in another environment or context. And that's where, when like Khmer talks about it, it's very important to both be able to use these tools like selfing that will put us down very intensive inbreeding channels, while also maintaining overall allelic frequency in populations, so that in the future, when unknown things happen, as they are guaranteed to happen, we will have variability that can adapt to these new environmental systems or dynamics. So selfing good, and also open pollination good. That's one of the things where it's like, next slide is exactly that. Like, random mating occurs when, and we kind of talked about this with the Hardy-Weinberg equilibrium, each member of the population has a chance to produce offspring, and every female gamete is likely to be fertilized by any male gamete. In that situation, allelic frequency will remain the same, levels of heterozygosity and homozygosity will remain constant, and genetic relationships will also remain constant over every, you'll get an inbreeding coefficient of 0, essentially. That is the best breeding system for maintaining a wide range of traits and all of the genetic diversity. It's not a way of improving a population for a specific select trait.
Okay.
And like, that's what we did for years. Like, it was random open pollination, maybe a little bit of skewing based off of, like, I chose a subset of my population, but I let like X number of males and X number of females make seeds, and then use those seeds the next year. That's a somewhat constrained version of open pollinating. It's not the true perfect Hardy-Weinberg, because you don't have a large population, but because you have multiple different pairs in the grouping, and because it's randomized within that group, you're maintaining a huge amount of diversity within that population, and you're not doing any positive selection one way or another.
So would that be a way, if you had a bunch of different cultivars there, would that be a way to kind of open things back up?
Yes.
Okay.
Yep, exactly. That's like, when Kev's been on a number of the roundtables the last couple weeks, and he's talked about his work with the Afghan population that he got, I think, from Indian Landrace. I could be wrong, but Indian Landrace Exchange is super awesome people who are out there collecting in Afghanistan, throughout South Asia and Central Asia. And he's taken the seeds that he has and he's just doing an open pollination with them just to maintain that population. And there used to be like, this was one of the other forms of like massive fighting in like, you can find old forum posts of people being very upset that breeders were breeding and going down the rabbit hole of selfing or backcrossing or intensively inbreeding varieties without also doing just the broad open pollination, because by doing that, that's how you get down into the bottlenecks that you're seeing. Like when I went to UC Davis, the professors basically, when they talk about breeding for pest resistance in most of the traditional crops nowadays, a lot of times they start the discussion with genetic engineering because they're like, well, fuck, we've already exhausted the germplasm. We went down these bottlenecks, lost a lot of the diversity, and now we don't really have any, like, there's just nothing, and like we checked and there's not resistance in the population for this thing. So now we're fucking creating these new GMO crops just to find some resistance. And that's why this right here of, even if it sucks and it's crappy and it doesn't produce like a commercial or a, like, this is the cannabis I want to smoke, it preserves that genetic diversity that is going to be the thing that gets us different traits that we need, that we don't know we need right now.
The starting point. You always can go back to the starting point. Yeah, definitely non-destructive editing, basically.
Yeah,
to bring it back to a music term, non-destructive editing. Yes.
Yeah, but no, that fully, like, that conceptualizes what we're talking about here, is that like we want to do non-destructive editing. We want to get that first, like, it's there, and then you can fucking tinker and go down from the different lines and the different variations and create bottlenecks. And the bottleneck isn't bad in and of itself. The bottleneck's only bad when there's no way of going down another route or having more diversity. And you can't always get more diversity within a population if you've already gone down the bottleneck. And so like when Wilhelm Johannsen went down with the pure lines of peas that he was making, he was like, well, fuck, I could cross these two peas in this family together, and I'm not going to get any increases in yields or variation in my trait. I'm stuck. Like, there's no more advance I can do. If I want to get any more increase, I'm going to have to get that genetic variation from somewhere else. And then, so the other assortative mating systems are, there's genetic assortative mating, and this is when you're doing inbreeding. This is selfing, full sibs, half sibs. To be clear, a half sib is when you take one parent and breed it to a lot of different things. So much of cannabis right now is half sibs. Like whenever somebody releases a line and it's like, we took this male and crossed it to 30 elite cuts, all of those offsprings are half sibs.
Yeah.
And so, or I mean, inbreeding with them is like breeding with a half sib. And so backcrossing is another very common example, and like lots of people do backcrossing, but with each one of these, you're going to have a different inbreeding coefficient. So with selfing, you're going to have a 0.5 inbreeding coefficient. With full sibs, it's going to be a 0.25. I think it's a 0.8, or excuse me, 1/8. So, fuck, I suck at, like I said, I suck at that. But it's like
we have calculators.
Yeah,
These days we're lucky. Yes. Yeah. I'm sorry. Go ahead.
No, totally. That's I cheat all the time when it comes to numbers. So according to and I would encourage anybody who can get this book. R.W. Allard's Principles of Plant Breeding is an absolute banger. The dude explains this shit in a way that is incredibly clear and concise, and he does it in a way that like it really synthesizes all of the history of our relationship with plants in like since we've been cultivating them. So it's really helpful. And but he says genetic assortative mating thus serves to reduce heterozygosity, increase fixation of alleles of the same locus, and when combined with effective selection, promote fixation of favorable multi-locus genotypes, as well as stabilize phenotypes to the extent that they are under genetic control. So when you look at THC, like you can see that we've done that. So through genetic assortative mating, and this is more, we'll actually get to this one on phenotypic assortative mating, but like we've gone down that road and really stabilized that phenotype, or that chemotype, as a lot of people will like to refer to it, throughout a lot of cannabis at this point. And we've done that through a form of either backcross breeding or like sort of adaptations of these ideas. And then inbreeding without selection produces increasing variability between families. So essentially, here he's saying that when you inbreed, you're going to create this extreme over here for the recessive trait, this extreme over here for the dominant trait, if we're just using traditional Mendelian sort of like dichotomy. And that's going to just be divergent. And without selection, you're not actually doing anything but creating divergence. Once you start applying selection, then you start driving genetic and phenotypic similarities that are essentially what you can see like through the mass selection process that we've been doing with cannabis. We've driven it towards a particular trait architecture through continual selection. And by everybody basically breeding the same 5 to 50 plants over the last 30 years, they're all a mixed population with relative rates of inbreeding that have undergone this sort of assortative mating. So we don't have a population that's in Hardy-Weinberg equilibrium, but we don't have an inbred population because we've just done this. Let's select that one. Let's intergress this new one. Let's do half sib breeding. Let's do backcross breeding. Let's do selfing. Like, because it's all of these things done by different people, and it's all this collective gene pool with relatively few starting points. It's all inbred to a certain degree, but it's all still very heterozygous and diverse because it hasn't been just intensely put into any one of these methods. Like, I've looked at a lot of different breeding, and the only person I've seen that has done like S4, S5 seems to be GW Pharmaceuticals, Hortifarm. If you look at their papers, they're talking about S5s in some of them, and that's like the most intensive inbreeding that I've seen. On the commercial market, you'll see S1s for sale, which are, ostensibly an F2.
Right. They've got huge R&D behind them now, and yeah, Jazz Pharma or GW Pharma.
Yeah. So exactly, where they're like, what they've done is like they've gone down these, and I'm sure I'm sure Oregon CBD, people that are in the like hemp space and breeding for those sorts of agronomic traits are really doing this type of work. But in the like commercial, you go onto a seed bank, like we're dealing with a population that's been subjected to a lot of this different variation in terms of like these different breeding systems with a set of a few, like, a handful of cultivars. but nobody's gone as intensive. So we've kind of got this in a patchwork system, but it's honestly based off of the next form of breeding, which is phenotypic assortative mating. This is what we've done. It's basically where you breed like to like on the basis of phenotypic expression. And so you pick the cookies and the ChemD, you're picking two high THC, high potency varieties. And the variation that you have there is more in like color and those sorts of expression, but they're both relatively similar in terms of their chemotype profile. And so when we keep breeding OG, ChemD, cookies, or all of the chems, and we take the things that were before them in terms of the Northern Lights and the skunks, which probably play into these more recent cultivars that we're using now, we've just been selecting for like to like phenotypes. So, those same half SIB lists of seed releases that people are doing, they've largely been selected on the basis that this is a high potency variety, and here's every other major high potency variety that's in circulation and being grown commercially, and I'm just going to cross them all together. And that process of constantly making selections on that basis drives extreme types. And so you'll really see a bifurcation in a population. And this is something that we've seen a little bit with like the CBD varieties that were saved. Like they've got an extreme CBD, the ACDCs, the Cannatonics, things like this. They're over on one side, and if you just continually select based off of phenotype for high CBD, or if you just base your selection off of high THC, you'll create a divergence between the two, whereas if you cross them, you're going to get more of a variety in between, and the intermediary types will express themselves. But when you just go to the extremes and pick like to like, then you're going to make it so that you have a vast divergence between the phenotypes and no intermediary expression. And that's kind of what we did to a large extent. Like some people maintained fixed or like those mixed chemotypes, but not really. Like those mixed chemotypes were really oh shit, I found a CBD variety that was in the population and hadn't been bred out, and now I'm going to cross that with a high THC variety that's in my, repertoire. And that's where we got a lot of the mixed chemotypes. And so this right here, these are the two forms. Like this is mostly what we've done, but we've done it using these different assortative mating systems, be it selfing, full SIB, half SIB, or backcrossing, at least on the commercial market. Like we're doing this selection process in these breeding methods. And yeah, so we've gone over inbreeding coefficient, these different systems. Then here's where we get into like actual breeding programs. So mass selection, this is the oldest form of breeding. This is something that everybody's done forever. And that's what we just talked about, where you're doing phenotypic based selection. There's a positive form and a negative form where you're actively positively selecting for good traits that you like. THC, yield, flowering time 8 to 12 weeks. These were the traits that we were currently selected for in the cannabis population, at least in drug cannabis breeding. Negative selection is like, okay, here's an off type, or here's a runt, or here's a plant that isn't producing the phenotypes that we're positively selecting for. Negative selection is just the elimination of those entirely from the breeding population. And so that's something where, you'll see negative selection was very popular with regard to like auto traits. People used to respond, like if you had a line that was expressing auto flowering, people would just be like, no, we're going to cull that from the line. Or negative selection against like CBD varieties and positive selection instead for high THC varieties, at least in drug markets. Then, so this is just basic breeding. It's not really systematic. It's just saying we're going to select all of the phenotypes that are good. We're going to reproduce them and use them for the next year. This is literally how plant domestication happened and how people bred plants probably for about 10,000 years until the 1800s. Pedigree method is a system of breeding where records are maintained on the individuals in the population. This was first developed in the 1840s and 60s by a seed company in France. It was a family-run company known as the Vilmorin Seed Company. They introduced genealogical selection, that's what they called it, or the isolation principle. It was later termed pedigree selection by F.P. Hallett in the 1860s. He was an English plant breeder guy. And the Swedish Seed Association in Savlov, they really like took this shit to the next level and collected extensive pedigree records on tons of crops for years and years and years. And that data set was really helpful in Nielsen L demonstrating the quantitative traits are inherited according to Mendelian inheritance. H.H. Love in 1922 first described pedigree breeding and bulk breeding in the United States. And this is the key difference. This comes from Georgia Qua, Principles of Plant Breeding and Genetics, or Genetics and Breeding, excuse me. A key difference between pedigree selection and mass selection or pure line selection is that hybridization is used to generate variability for the base population, unlike the other methods in which production of genetic variation is not a feature. So this is the key distinction, is hybridization happens in pedigree breeding, and it's a way of creating variation, where the other methods are really just methods of pure inbreeding going down the line. This system is illustrated here in this chart by, it's a paper by Robert Clark and Mark Merlin, but it shows the pedigree chart and the histories of, supposedly, of the breeding of skunk number 1. And it shows that at first you have a hybrid cross between Mexican, Afghan, and Colombian. The number 1 female and the A male were selected, and then the number 13 male, the number 2 male in a subsequent generation, and so on and so forth. And you can literally track the males that were used in the pedigree for the creation of what became the Skunk 1 line, and how after supposedly 5 to 9 years of inbreeding at like F9 or something, they then started doing their yield tests and their combining ability tests. So this is where they then took specific plants, crossed all of them together, and saw which combinations between which males and which females produced the best end results. And that's how supposedly they created the Skunk 1. I keep saying supposedly because, like, there's all kinds of controversy there, and people say it was done this way and that way. I'm just saying that this chart shows how they claim it was done, and it definitely shows, like, this is how you can do comprehensive pedigree plant breeding and maintain these kinds of records and show the lineage of a variety that you breed. And that if you go through this type of method, that this is how you're going to be able to produce the inbred lines that could then be used for create as breeding stock for a hybrid breeding program or for something along those lines. And that's you know, what they've done and what they've demonstrated. And, you know, Robert Clark and David Watson and Hortifarm, for better or for worse, regardless of whatever people's opinions of their politics or like business operations are, like they've proven that you can take cannabis, apply these breeding methods, and produce a very valuable product line that can then be monetized and put into like the pharmaceutical pipeline. And so, you know, I'm not putting a value judgment, good or bad, on that. I'm just saying that like value judgment on like they were successful in doing what they like set out to do, and have proven proof of concept. And so we can all learn from that, at least, even if we're like fuck pharmaceutical companies.
Yeah.
Yeah. And bulk selection is similar, but bulk selection is where you essentially just maintain a much more open and diverse population. So it really, you can see here in these generations where you have bulk plots and very limited selections being happening in these early plots. You just grow out 1,000 F2 plants, you get seeds off of them, and then you take all those seeds, you combine them all together, and you do that for multiple generations. And then once you're at like F5, you start doing selection. And the reason for that is this was a breeding method that was applied to barley and was also part of a massive, UC Davis did a really, really long, very extensive breeding program with composite crosses. And it's also been known as evolutionary breeding method, where when you do less selection early on, you allow natural selection to have an impact on the direction of the breeding program. And that makes it like, it's useful for creating adaptive varieties, and it's also useful for maintaining a bit more genetic variation within a population, so that you don't cut off different avenues that you can go down in terms of the breeding program later on. So this is another way of kind of melding those two ideas of maintaining and capturing genetic diversity while still at later points doing intensive selection. And that's what you see more down in the F5 to the F6 generation, where here yield trials are being done and selection is being done on those individuals that produce the desired outcomes. This is something that you would want to do if you were breeding crops specifically for a particular place. So if you know that your cultivators are growing in this, and like you said, tents, like, you know, you would
Right, yeah.
You'd want to like make sure that you're doing your yield and your test grows in tents. You wouldn't want to be doing those out in, you know, massive field grows, because a massive outdoor field grow is probably going to have a lot different set of variables influencing on an environmental level that are totally independent of the breeder's selection, the outcome of the genes and the phenotypic expression, but more importantly, the genes in this instance. And that's the thing where, you know, it's really important to breed in your end environment. If I'm an outdoor breeder, it may not necessarily be the best situation to take my stuff and grow it indoors in certain instances, because of the different environmental pressures that are happening outdoors, those that are happening indoors.
Yeah.
indoors light leaks are such a huge issue, and breeders are oftentimes, hopefully, selecting against those plants that are more susceptible to light leak issues. And so that's, just an example where this is something that is a breeding method that captures some of that natural environmental selection and helps to influence the population dynamics that way, as opposed to just, an individual person doing artificial selection that may not correspond to environmental dynamics. Pure line method is the form of breeding that Wilhelm Johannsen really helped to establish, and it's just selfing. It's taking an individual line, and it could be a mixed line, and then you would select the individuals within that and self those and get them to a state of pure homozygosity. It's really just it's the simplest, most straightforward process of inbreeding. You're just selfing your line. You select an individual plant, you self it, you take its offspring, you self all of those, so on and so forth down the line. And that sorting process will eventually leave you with essentially two homozygous distinct lines. That process got added to by the idea of single seed descent, and this takes it one step further. It's a way of really rapidly doing this. I'm not entirely certain, but I've heard the Oregon CBD people talk about rapid breeding advancement, and I think this may be the method that they're using. The idea for single seed descent is that in an F2 population, you take one seed from every plant and you do that continually for multiple generations. You're going to rapidly increase homozygosity of every one of the possible homozygous combinations that you can create in the population for both quantitative and qualitative trait inheritance. And so this way, you're not going to lose any of the possible genotype combinations. And it's really the fastest way that you can move an entire population to fixed homozygosity without losing allelic frequency or like specific alleles that could be made homozygous.
That's pretty darn amazing. I'm gonna come back. Well, I'm gonna come back to all of this, but
Yeah,
that's fascinating.
This theory is really something that took plant breeding, and like so much of plant breeding is based on shortening this.
Right.
Like this, 10 F10, like theoretically, if you're an outdoor grower, that's 10 years. Assuming no artificial environmental manipulation, and so that's a fucking long time. And so plant breeding is all about how do we advance our goals and our objectives in the shortest amount of time, and all of this math and all of these methods and principles are really just about reducing that time frame.
It's interesting. Okay, I'm seeing the answer here in the slide. I was thinking, okay, you said a single seed from each generation. Does there have to be a male? Is it the same male? No. Okay, I see here that it's self. So every F2 through F5 generation, you self that and then take the single seed to kind of follow the roadmap here.
Yeah, and in cannabis, it's complicated because essentially, in reality, you would have to do a clone and a side-by-side, so that you could have your clone that you're for seed production, or you do your plants for seed production, and then you'd also do your plants for testing and seeing how they produce as phenotypes. So every one of these, because we don't have a selfing variety or a selfing species, and we have to do it artificially, essentially what we would have to do is run a trial with all of these plants grown for production just to see how they express in a production context, while also doing a selfing on the side to then do this breeding. Or if we don't give a shit and all, like, we've already decided like variety and A and B are the shit and we're gonna fucking go down this rabbit hole, then you would just be, you have your bulk population, yourself, 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 look at what's in there.
Huh.
And that's really the idea here, is you're just trying to create a population that's as inbred as possible without losing the frequency or individual combinations that are existent within the population. And once you've gotten all of them homozygous, roughly speaking, you know, we talked about residual heterozygosity, but the vast majority of the lines are going to be homozygous by F6. And as a result, you're then making your selection, like, let's just say this corner right here is the like 5% threshold. Well, like, you're selecting over here, so to speak, you know, for that like 5% or 10% threshold of the most ideal, best in the population. And if you had transgressive segregants that were emerging here or in any of these previous generations, you're not going to be losing those transgressive segregants by accidentally crossing them with something else in their population. You're going to keep those as a constant in that population each time. And then you'll be able to track and identify, like, fuck, you know, we took this plant, we selfed it here, and in each one of these generations, when we look at the test plot, it's expressing on the, like, you know, the 5 to 10% in the extreme or exceeding the parent lines. Clearly, this is the transgressive segregant for whatever the specific trait of analysis is. So we're going to select this one, and this is the one that is the inbred breeding elite that we're going to be using once we go to commercial production of, like, making a hybrid outcross with it. And that's when you get into your doing test cross. And, you know, on the previous chart with the skunk #1, where you test your specific combining ability and your general combining ability of your new variety that you've developed down here that's super inbred, you then hybridize it and see what the yield of the offspring are. And the pairings that are the best end up being the ones that become commercial offspring or commercial offerings.
Interesting too. The thing that I always get curious about, and I don't know if it's even part of the equation here, but a lot of times we'll hear, as you inbreed it further and further down the line, though, you're losing the vigor.
Yeah.
So is it, so in the goal here to reach like the F6, the F10, is that something that's even accounted for in the equation, or is that, you know, not part of the goals on the list?
So it is, but the way that it's evaluated to a certain extent is by looking at the parents and then the F1 offspring. So if you have variety A and variety B, you're going to look at what's the yield data of variety A, what's the yield data of variety B. That should be a known quantity before you start. Like, for inbred varieties of corn, like Mon 187 is like a variety of corn, and then B something or other, it's got a number. Like, those are two inbred lines that are used in a lot of breeding. When they cross, like, they're of known yield variables. When they cross that offspring and the hybrid offspring that results, it then has a mean yield that it produces. And then every one of these generations, if you're doing the test plot and actually looking at the production in a commercial sort of setting, you're going to see that the yield mean, you know, you're going to have your mean of the population, but then you're going to have your extremes in the population, and those extremes are, that's what you're testing each generation. And so even though you're getting a lower and lower and lower yield, most likely, unless you're dealing with a transgressive segregant, you're still going to be able to test those yields back to the relative, to the previous generation. And then once you outcross it, then you'll be able to test it back to this F1, this parent, and say, okay, we inbred this line, we then crossed it with this other variety, and that cross produces a yield that exceeds the F1 hybrids and the parent material. So is now technically an improved line. And like, that's what they did with corn. Like, every time they got another dot on that chart showing increased yields and like different varieties that came online, that was the result of them basically being like, okay, we can reach this point in corn yields now. Let's make another inbred line and select for trait or for yield and improve upon what we've already developed. And they were able to do that, you know, in a linear fashion. I think that's what that is. Again, no statistician here, people. But yeah, so they were able to create incredible gains in corn yield through this type of breeding method, where they would inbreed it intensely and then outcross it. And then trend, to the extent that that's kind of like that rule is broken, it's with those transgressives, because even though they're fixed and have a homozygous, like, they should be suffering from inbreeding depression because of the because of epistasis. And epistasis is a very complex, like, gene interaction thing where all of the genes can produce particular results, even though there isn't necessarily like a change in the genotypes, but all of the genotypes in a population could be relatively similar, but you can still have some variation that happens as a result of epistasis. This is an idea that complicates Johannsen's idea about pure lines, but it still works together, and it's all because of the linkage between different traits and their complex interactions on like on a quantitative level. And so epistasis is something that can end up making it so that you can get epistasis and complex gene interactions produces transgressive segregants, which, even though they're homozygous, still express traits that exceed their parents and don't necessarily conform to inbreeding depression. And this shows backcrossing, and this is like a breeding method that we've kind of discussed. It really shows, like, you have your recurrent parent. This is your princess in I believe that's what Mr. Soul referred to his keeper cut that he found as a bagged seed. And then he had a pollen donor. He made his F1. Then you take that and you cross that F1 with the recurrent parent, and you've theoretically recovered 50% of the genome. You backcross it again, theoretically recovered 79%, and so on and so forth, until eventually you've got 92 to 99% of the genome recovered. They've added a selfing here at the end of it, and the reason why you would do that is because by selfing, after you've done a lot of backcrossing, if there is heterozygosity within the population, by selfing it, you're reducing the overall heterozygosity that you're going to have within that population, and you're going to create a much more uniform and like less diverse population by getting rid of that residual heterozygosity and the the variation that isn't gotten rid of through backcrossing, because backcrossing doesn't eliminate all of the variation. If the recurrent parent is heterozygous, then by backcrossing, you're not necessarily moving away from that level of heterozygosity. It will constantly be a feature of the breeding program. And so by selfing in later generations or between each backcross, you're essentially developing a way of getting rid of that level of heterozygosity and increasing the amount of fixity in your genotypes. And just for like the main reason backcrossing, Chimera talked about this. In cannabis, people do backcross breeding for a reason that isn't really, it's not the way that scientific plant breeders use backcrossing, typically speaking. Backcrossing is typically done to introduce foreign genes or introgress foreign genes or traits from one line into another. So you've got a commercial variety that is really good for all these different reasons, and it's got success on the commercial market, but it's powdery mildew susceptible, or it has some other agronomic trait that makes it undesirable. If you have germplasm that has the desirable trait, you want to take that desirable trait from this other perhaps undesirable germplasm and put it into your commercial variety. Backcrossing is the way to do it. And they call that introgression. And the first time it was done in plant breeding, even though it had been practiced in animal husbandry for years, and the genetics had been the inbreeding coefficient and different things like that had already been worked out. It was used in plant breeding in 1922 by H.V. Harlan and M.N. Pope, and they wanted to take a strain of barley that was, I think it was like rough on, so like they had rough seeds, and they wanted to make it smooth, or vice versa. Either way, they took this variety of barley and they tried to breed the trait just using traditional mass selection, where they were, F1, F2, F3, F4. They tried bulk selection. They did all these different inbreeding using sibling and selfing and different techniques like that, and they couldn't get to a stabilized expression of the trait in the population. So they decided that, well, we have this plant that has the trait that we want, and we have our commercial variety. Let's just take that plant that has the trait, cross it to our commercial variety, and instead of doing sibling or selfing F generations down the line, let's backcross it to the parent plant of the commercial variety that we have that's running. And so they did this process, and they were able to then create a, let's just say, smooth-on variety of barley. So it no longer had the rough seed, it now had a smooth seed, and that was a trait that they were going for, I believe. It was one or the other. And that proves the method that you could take a trait that was unresponsive to traditional breeding methods and use this backcross method as a way of breeding or introgressing that trait into the genetic background of the commercial variety. And now the way that it's really used is in disease and pest resistance breeding, and or genetically modified organisms. Backcrossing is essential in that breeding process, but really what they've done is, and that involves a whole bunch of obvious genetic modification and stuff like that happens on a molecular level prior, but then in the actual field, the process of breeding that trait into a population is done through backcrossing. And that's really you don't see plant breeders saying okay, I've got, this variety, and I want to now put it into seed form. So I'm now going to do a Backcross project, which is kind of how the whole idea developed in the cannabis world.
Okay. Yeah.
So yeah, those are the basic principles and concepts of breeding. I had some other stuff that I wasn't able to throw in that really gets into the more advanced, like there's the breeder's equation and the actual statistical methods for doing a lot of this stuff. And like the key thing though is that with all the stuff that we've talked about, if you have data and these methods, you will produce the results that have been experienced in every other crop. And you don't need millions of dollars. You don't need a scientific degree. All you need is an interest in the subject matter, a willingness to dive into it and apply the concepts to this plant that we find super fucking awesome.
Well, this might just make it a good breakpoint then. I mean, I would definitely love to keep picking this up and keep going. I know that I need to go back, and hopefully a lot of other people, will go back and review some of this material and just wrap our heads around it a little bit further. A lot of the conversations that we hear on a daily basis, we participate in, a lot of that is filtered into these topics and these concepts. So I love it and I appreciate it so much that you put these together. And yeah, I would definitely love to keep going on it. At some point, if you're able.
I'm down to come back and to dive back in, and there's definitely like all of the next, essentially the next section that I had like at least mapped out is really just the application of all of this stuff in a more specific and like detailed way. And I feel like, it's totally a good point to take a break, but if people want this one, like the slideshow presentation or whatever, I'm going to put that in the file that I've shared with you. And
awesome.
On that Dropbox, there's a Google Drive Dropbox. I've got a file that Chad and I have shared, and we'll share it with like the listeners and people who are interested in this. And there's the thing about like, I was able to do this because I put together that Dropbox. That Dropbox has all of these ideas, all of these papers, and all of the material. And like, you can just dive in. And if there's a part of this that you found useful, you can get into this slideshow, look at that, and then go into the file and the Dropbox or the Google Drive and really dive into these subjects deep and get an understanding for them. There's a glossary and a dictionary that has all of these terms defined. It's in the textbook section. There's like there's multiple introductions to plant breeding, to population genetics, all of these subjects. And so, even though it may feel like daunting or kind of like hard to get your access point into it, this material's all been organized and available. And I'm down to engage in these discussions.
Just start somewhere when it comes to those folders. And I mean, nobody listening right now that hasn't looked at these folders has no idea how big of a gift this is to everybody. I freaking just jaw dropped when I got in there, and I'm like, I'll be busy for the next 20 years. It's awesome.
It's really like granted, I have not read something. I want to be clear, it's not like I've read every one of these papers. I just see a paper, I'll read the abstract, or I'll find books, and I'm like, this is useful for my weed shit. And like, it was just a huge Dropbox file that was disorganized for the last several years. And then I was just like, fuck it. Like, I have to get some semblance of what's in here. And when I did that, yeah, it's just made a lot of things. Like, I was able to put this together while doing a whole bunch of shit this week. And it was easy because it's all just there. It's like, you want to learn about backcross breeding? There's a section on that. You want to learn about, you know, the biggest thing I feel like is the mystery is this idea of quantitative traits. And if people just dive in on that and get past the Mendelian P thing and get into the, like, how is yield actually a trait that we can improve on, that's where we're going to see just enormous gains in breeding.
And this is the right time again. You know, it's still such an infant mainstream industry, and I'm not talking about, you know, mainstream as in big companies. I'm talking about mainstream as in the sheer number of people that are doing it these days. So we're at a good point to start off right, set the record straight. I mean, we could go down the rabbit hole inevitably of you know, markers and different traits that might be put into different things, and how we need to, you know, open source breed now, or, you know, get not the galaxy, so, something similar to the galaxy, you know. That's always there. But yeah, I we'll definitely keep this going. I'm having computer issues now. Like, I still see the bulk selection slide over the screen, but faintly behind that I do see us. This is kind of a every now and then recurring video card issue I have. So I can't really see chat, unfortunately, because I'd love to get them some of their questions up for you, but that in itself just makes us, we'll have to do a part two, so everybody can save them up if they can't, you know, go back. I'm gonna go back. I'll watch this again, and again, poke around in that directory. I just, you know, my first thing I did is genomics. I wanted to learn more about it. There was, I think it was like 496 page textbook in there. I said, okay, I'm starting here. So start.
No, totally. And I'm down to do, like, you know, you reached out and were like, hey, I want you to come on and do genomics, and like explain it in the simple terms and tell people how, like, with pencil and paper they can do this stuff.
Yeah.
And I was like, well, fuck, that's literally like the whole fucking story. And like,
can you just A to Z this in, you know, 30 minutes with commercial breaks for me, please?
Well, yeah, and it's like all of the genomic stuff that we have in terms of markers. Like, you guys did it great the other day, where you defined what a SNP was, and you got into like the real specifics on, like, what's a genome-wide association study, what's a SNP. And I was like, after I did all this, I was like, fuck, I haven't said SNP once. There's no markers, there's no like, this isn't genomics, quote unquote, but it's what you need to do to be able to do that without those things, in terms of like the high-powered computer technology that runs genomics. Was, you know, a 19-year-old in a fucking doing his homework and being like, well, fuck, like, I think there's a link here between these different traits and their inheritance. And like, that's something that, you know, I'm totally down to come back on and talk about SNPs and like actual the very specific, like, because all these different markers are used for different things, and some of them are really good for doing, like, you know, what Phylos or medicinal genomics or some of those people have done, where it's like phylogeny, like, what's the associations between this population. Some of them are really good for being like this trait. Some of them are really like economical and like affordable, but don't have like the throughput capacity, or some have a massive throughput capacity but have limitations on the length of the reads that you can do, and like all of these different variables that I'm down to talk like genomics
Yeah.
as well as this. Yeah.
Yeah, because I would definitely love to get into that as well in the future, because I think that is gonna kind of, dictate a lot of the future of what we may see on the market, not only in the flower, but also in the seed market in the future, because this work is being done. I mean, we've already mapped, like you said, certain alleles for THC, for CBD, certain pest or IPM resistance.
Yeah. Yeah.
Those things are already being worked on. But for somebody in the majority of our situations, we need that foundation. I like the big picture, but we also need the foundation, and this is where it all starts.
Mhm.
So
Yeah.
I'm glad that we did kind of go this route, and that's what a lot of people here are interested in. That's why they love the topic, and I'm no different. So yeah, well, we could definitely expand into that too, because I'm always curious. I've got a million and one questions for that, but for tonight, you did a great job of actually answering. I have a bunch of things written down here, but as this went on, I knew to hold my questions to the end. But yeah,
Well,
you
no, I
kind of nail them.
appreciate it.
Yeah. Is there anything that you kind of wanted to maybe mention that I didn't ask you, or that you didn't get to tonight, that and sometimes the answer to this is no, but if there was anything that you thought you might have left a cliffhanger for somebody, or just good resources. You've already put a few out there, but I'm gonna punch in your, oh god, I'm gonna try to punch in your Instagram here. Let's see if I can.
Yeah.
Oh, my screen is so horrible.
Well, one thing about the if people have questions about the Dropbox, it's I don't think I got the chat up, and I don't think you need um, to have a Dropbox account necessarily to be able to access it. I think you can access it without having an account. And there's a torrent. I don't know anything about these things. I remember they gave computers viruses a long time ago. I don't know how to use it, but I know that Frackler on IG is a homie, and he was like, "Hey, send me your like Dropbox file, and I'll put it up on this torrent thing, and everybody can like crowdsource, download, and do cool distributive network sort of stuff that I don't necessarily understand." But there's that link, and there's also a Google Drive link. So if people have issues, hit me up. I will try and help you get the access to it. And I just want, like, it's purely education. Like, I want everyone to be able to access this information. I also encourage everyone to, purchase these books, like, you talked about finding a old soil science book for $4. Like,
Yeah.
I spend a lot of time, like, my absolute favorite breeding book is not digitized. I referenced it before. Principles of, there we go. Principles of Plant Breeding, Robert W. Allard. This is the second edition. This book is, sorry for the glare, it's Cron. It's the best plant breeding book I know of. And I have multiple other open source plant breeding books that are from college courses or that are available online, and they're great. They're very informative. I don't know how dude does it. Like, he explains these things in a way that is incredibly intuitive and clear. And he also, you can find tons of papers by Robert Allard online. He did tons of breeding at UC Davis. The work that they did was pretty interesting. Coit C.A. Sinison, I think that's how you pronounce his name. He was a guy who really developed evolutionary breeding method, and it's cool to see somebody who's tracked a population for, I think he got up into like F100 or some shit. Like, yeah, no, fucking crazy. And the amount of data that he tracked on this cross and this population, and Robert Allard does a lot of work with the same populations and like demonstrating in different graphs of like the inbreeding dynamics that happen with it. And so that's, I would just encourage people to seek it out. And also, if you ever have the opportunity, go to a university extension program.
Mhm.
Like you don't have to have a degree to go to UC Davis and do their plant breeding program. And I assume
awesome.
because of COVID, it's probably online these days.
Right.
But that's something that is really helpful. Like, I was given the tools and the ability to understand a lot of these ideas about genomics and stuff, and like what a SNP was. I remember during the whole Phylos debacle, a grad student from a university who was like doing work and looking to get into the cannabis industry after his academic thing, was like, "Do you even know what a SNP is?" And I'm like, "Bro, like, one, I've got Google, so like even if I didn't, I'd fucking Google it. But like, two, like, yes, I fucking know what a SNP is." And then like said it back to him, and he was like, "Oh wait, like,
Right.
maybe you know something." And it's like, that's something where I just feel like having the language and the lingo is really like, it's very technical, and it like, you got to learn all these acronyms and shit, but it's worth it, and it empowers you so that when people do shady shit and try to run a scam, because a lot of like genomics right now to a certain extent is like, here's this shiny new tool, here's all the bells and whistles, we can sell you this product that is a lot of fucking money, but like, what's the practical outcome? Could I just do a simple math equation and hang out with my plants and get a better result?
No, you're not wrong there. You're not wrong. And yeah, that's for, the show that we did the other day where I talked about the single nucleotide polymorphisms.
Yep.
Uh, I'm like looking, I'm like, where did I write that down? But yeah, I had to look that up because that was in the header paragraph of the study we were looking over. And it's like, I probably said it last night. It's like, I know how to pronounce this word, but what does it really mean? And so, yeah, that's why I looked it up. And SNPs is a much easier way of getting there. So when you do know that term, you don't need to go through the tongue twister, and you can just keep going through it. So again, to reinforce the point of terminology being pretty handy.
Oh yeah, and SNPs, everybody loves SNPs because it's like, simple, you can just call them SNPs. Fucking RFLP, like, fuck that shit. Like, nobody wants to talk about those markers. Like, it's not fun. Like, what the fuck is, like, it's in there, and I'm dyslexic, so I maybe like superimposed something, but like,
Oh no.
it's one of the markers, but I
Right.
like can never fucking remember what the fuck it stands for.
Yeah.
And, yeah. So, yeah, and another couple more things. Gnome de Fume, Coop is on Mr. Nice Forum, but like Gnome de Fume on Instagram, he's really the person who I got that, like, who told me, like, he kind of disabused me of the notion of genomics as being like the be all and end all, and was kind of like, fucking do your maths, bro, and pull out a paper and you can do it. And he's somebody who I encourage everyone, like, follow him. I'm sure he doesn't want me blowing him up. He's kind of like a low-key cat, but like, dude's fucking brilliant. And I just saw Bordeaux. Did I say borderline or Baudelaire? I don't know. I thought I saw in comments Baudelaire. I'd say shout out to Baudelaire. He's a longtime overgrow cat.
I'll tell you, like, if you guys can see my phone here, that's what my screen looks like right now. I don't know if that's focused, but yeah, I'm kind of hosed. I can't see anything that's going on, really. I can hear you, and I still see a few buttons. So we can keep going.
Well, yeah. Uh, restriction, fragment length, polymorphism, chimeras dropping science in the comments. So like,
Oh, awesome.
fucking everyone go follow Breeding Cannabis. Breeding Cannabis, Chimera. He's really, if you, he should be on here. Like, I mean, the guy is like, he is.
If he's in the comments right now, heck yeah, because he's another person that I've really enjoyed listening to on these breeder panels that have been going on in the clubhouse lately on the FCPO too.
Yeah, no,
So
his
yeah,
that was like huge moment when you came on the other a couple weeks ago, Chimera. That was great. Just loved listening to it and going back to it and listening to it
yeah,
repeatedly. And so there's people in this field who are giants, and we stand on their shoulders, as like is the common saying, and seek them out, like all of them, like. Or I don't care how we feel about them. Like, you know, I know there's differences of opinions on different people, and there's different camps, and like all of that is, yeah, it's one of those things. Yeah, exactly. And like knowing it is, and like going back to the, I, I did the Breeding the Culture series, or I'm doing it in part to understand the thinking and the logic and like what motivated these individuals, and trying to do it from an unjudgmental standpoint, and instead learn and understand and like sit back and be like a fan and appreciative of all these other breeders. And that's something that I feel like we, you know, you mentioned the tribalism, sort of like the dynamics of that we get into, and it's like it's so much, you know, it's good to understand people and try and like get out of yourself and recognize them for their own terms and their own merits. And
yep.
so that's something that I hope that the series can do, and that we can really like, you know, look at the different methods and compare and contrast without it coming off as being like judgmental.
Yeah, and you know, even if some people are judgmental about other, you know, I'm not a huge fan of it, but that doesn't mean you can't look past it to look at a particular aspect. And in this case, some of it is this, you know, the science aspect of it. So,
Yeah.
yeah, it's all for the better of everybody.
Yeah. And shout out to Kevin McKernan as well, who's been mentioned in the comments. Like the work that Medicinal Genomics is doing is incredible. If you can, his 2019 Medicinal Genomics presentation, he goes really in depth into their research into the Jamaican lion genome. And what's really cool about that is they went and they sequenced the male genome and really spent a lot of time focusing on the Y chromosome. And so because of that, like the Y chromosome, it's heteromorphic chromosome, so the Y chromosome is larger than the X chromosome, and so there's additional genetic material. It's not entirely clear
Right.
what that is. A lot of it's just repeats and like noise and non-recombining areas that aren't necessarily that important or significant, but like other parts of it potentially. And Kevin, like it would be awesome to hear him talk about it. But he gets into some of these specifics on like there are some sex-linked traits or some genomic
Huh.
markers that indicate that there could be good genetic variants on connected to the male chromosome. And but then there's also tons of other stuff that's just not relevant, and particularly like it's not recombining. It's
Right.
just part of the
Just
sex
it
chromosome.
filler. That's
Mm-hmm.
interesting because I did hear Mr. Toad talk about that or reference it, and also too, I did reach out to Kevin McKiernan the other day just as a kind of a hello, and he totally got back to me and said, "Hey, yeah, I've done stuff with Future Cannabis Project before, so that might be a possibility. I'd love to put some of these, other people's together. It's like
Yeah.
it's like kind of when you're talking to your hero and then your hero gets his heroes. It's like, well, yeah, bring it on.
So, yeah.
We'll
No.
start
That I
doing
mean,
it.
totally. Like, I feel like Kevin and Ryan Chimera, like, there are people in this field who, you know, as a self-taught, educated sort of person who was like homeschooled and went to like community college, like doesn't have a hard sciences background or a mathematics background, you know, I listen to people like them or Matthew Gates where they know the specificity of the usage of terminology. They're very precise and exacting, and they understand the material on a much more higher level than myself. And you know, I'm still don't let that discourage anybody. Like, go hard on that shit, and like, just because I don't necessarily have the background they do, I feel like beyond amazed and honored to have been able to interact with all of them and like see them as friends and colleagues in this cannabis space. So like, thank you guys for just, you know, for being peers. Like, all like the fact that you're doing a study, like the study paper reviews, just the format of that, the study group, that's what it was called.
Yeah.
Fucking love it. Like, I
you.
so much, like, anytime you want me to come on, like,
Hell yeah.
I'm totally down, because, you know, even if I'm not academically trained, I love reading scientific papers.
Yeah, and you know, you don't have to have letters behind your name to be an intelligent person either.
Yeah.
So yeah, I totally don't subscribe to I don't subscribe to that, but it is to your point about these people. You know, an analogy is people love seeing, you know, sports stars perform well. They love their craft. They love how they do it. They love that, you know, there's a difference between the minor league and the major league teams. Yeah, when you do get to talk to or listen to people like that, it's inspiring. It's like you're like, damn, I want to have that type of command of the information and stuff. So, yeah, I get where you're going, and it's, you know, like I said, man, it's been really fun listening to them and yourself, sir. So,
well, thank you. And yeah, that Chimera 100%. I'm totally down to plan of action, course of, like, that means a lot. And I want to thank Peter and you and everybody who's really dedicating themselves to spreading this information. And even people who, you know, I disagree with at times. Like, I feel that this thing is something that's bigger than all of us. And so thank you.
Yeah, my pleasure, man. And I really wish I could see chat, 'cause I feel like I'm missing something, but I will go back when my computer recovers. Damn video cards, damn Apple. I'm switching. I'm done. Yeah. Okay.
Yeah.
Well, let's wrap it up here with the promise of we're gonna be back, and I think we'll be back with an expanded cast, and we'll just freaking keep nailing down, keep driving down into it, because again, we're all about education, and it's great that we could come together to do it. So thank you for your time tonight, especially after your marathon day of driving.
Yeah. It was good seeing you today, Peter. I was down in LA, back up in Sac. Super stoked to be home. Yeah.
Cool.
And also got my homie shirt on, Santa Cruz Cannabis Co. Adam is a homie, and I hope he's watching, or will watch it later. And like, shout outs to all the homies, everybody who is part of this thing we do.
Yep. Well, right on, guys. And thank you, chat, for joining us, and everybody who's going to watch this on the replay. We sincerely appreciate that. And thanks for hanging out tonight. So with that, I will end the broadcast. Thank you, Trevor. Appreciate it, man.
Thank you, man. Yeah. Likewise, Chad. Take care. Chat, everyone. Thank you. You're awesome.